I hope yesterday evening was great and you are not so tired to have a power still and a lot of question in your head to have for our lecture. And first lecture will be presented by Wigmar Hugado, who will have presentation related to mangrove forest cover change assessment in Philippines.
Thank you for the introduction. By a show of hands, who among this room has ever visited a mangrove forest before? Can I see? All right, so we only have a few. I expect that. All right. For those who haven't heard about mangroves before, mangrove forests are among the most productive ecosystems on Earth, coastal ecosystems to be exact. But many biodiversity hotspots like Sibuyan Island has been understudied. And so this was the focus of our study, which we conducted in the Philippines. By the way, I'm Wigmar Hugado. I'm a faculty member at the University of the Philippines, Los Baños, and currently I'm on a study leave, taking my master's in landscape ecology and nature conservation in the University of Greifswald, Germany. So, this study is about analyzing or assessing the mangrove forest cover change of Sibuyan Island, Philippines using Landsat remote sensing. And before I go on presenting our results, I would just like to give a short overview on what mangroves are. So mangroves are basically plants. They are shrubs and trees that are found within the tropical and subtropical region. So here you can see these trees here are mangroves, and they naturally thrive in intertidal zones. So intertidal zones are where the ocean meets the land. So with this regard, these mangrove plants have become adapted to brackish water conditions, or kind of mixture of salt water and fresh water. So they are salt tolerant. So they are very adaptive. And just to point out here, in intertidal zones during high tide, it is covered by waters, and during low tide, it is exposed to air. So this is just an example of a mangrove species which is very common in the Philippines, Rhizophora mucronata. And here we can see the distribution of mangrove species in the world. And as you can see, they are concentrated within the tropical and subtropical band. And here you can see that most of the mangrove species in the world are found in Southeast Asia, approximately more than 50 species. And of the 65 known mangrove species in the world, half of it is actually found in the Philippines, which is my country. So this is the Philippines. It's an archipelago, meaning it's composed of different islands. 7,107, in fact. And so why is it important that we talk about mangroves? First, they are effective carbon sinks. They store about 4 to 20 billion tons of carbon globally. And not only that, they also protect coastal communities from storm surge and tidal waves. And they serve as habitats for faunal species, including mollusks, crustaceans, fishes, and birds. And they provide forest-based products and livelihoods for coastal communities. For example, wood for fuel, honey, and here in this photo that I will show you, so this is wine made up of nipa, which is a minor mangrove species in the Philippines. And it also provides thatches for roofing, and most of the mangrove communities in the Philippines also rely on fisheries and tourism for livelihood. So, this study was conducted in Sibuyan Island, which is a 44,500 hectare island in the Philippines, located in the Romblon province, and it is surrounded by deep sea. So here in this map, you can see the Philippine archipelago, and in the heart of it, you can see the Romblon province, which is composed of 3 major islands, and one of those islands is Sibuyan Island. Sibuyan Island is composed of 3 municipalities, Magdiwang, San Fernando, and Kahidiocan. So this will be relevant later as we look on the change of mangrove covers. And this island is known as a critical biodiversity hotspot because of its high endemicity and threats of loss due to anthropogenic activities. In fact, there are 700 vascular plant species recorded in the area, 54 of which are endemic to the Philippines, and 180 are native to the Philippines archipelago. 54 are endemic to the island. And it has never been connected to the mainland. That's why it hosts a very high endemicity because the species evolved independently on this island, just like the Galapagos Islands. That's why it is often dubbed as the Galapagos of Asia. But despite the growing literature in Philippine mangroves in general, only few studies has been conducted about Sibuyan Island mangroves. And in fact, prior to this study, there has been no conducted cover change analysis of mangroves in this island. And so this study was conducted, and its objectives are first to assess the change in the mangrove aerial extent of Sibuyan Island between 2010 and 2020 using Landsat imagery, and also to describe the mangrove vegetation density of the island using NDVI. And lastly is to recommend strategies for mangrove conservation of the island. And in order to achieve these objectives, we have used the Philippine Mangrove Extent Layer by the Blue Cares project to map the aerial extent of the mangrove forests, and this is complemented by manual digitization. And for the NDVI analysis, we have used Landsat 7 scenes for 2010, and for 2020, Landsat 8 images. So these images have a 30 by 30 pixel size and was chosen based on the criterion of less than 10% cloud cover. It is, the classification was conducted in ArcGIS, and accuracy assessment were also performed. So NDVI refers to Normalized Difference Vegetation Index. So basically, this is a metric that uses the difference between the near-infrared band, which healthy vegetation reflects, and red light, which vegetation absorbs, in order to quantify the density and the healthiness of green vegetation. So basically, you will have values ranging from -1 to +1, with values closer to +1 indicating a more healthy or green vegetation. And for the purpose of this study, 3 vegetation densities were classified, namely bare, sparse, and dense, based on these NDVI ranges, which were adopted from the study of Nesperos et al. 2021. And just to have a quick visualization of how this looks on the ground, so this corresponds to bare, sparse, and dense respectively. All right, so for the results, one of the key results of this study is that there was an increase in the mangrove cover from 2010 to 2020. So approximately 182 hectare increase within this 10-year period. And if we will look at the aerial extent based on vegetation density, so here we have the 2010 and 2020, so bare, sparse, and dense. So we can see that majority of the increase is attributed to the increase in dense vegetation. And in fact, dense vegetation increased 6% per year annually, or percent annually, and the bare and sparse vegetation decreased by 26.7 and 10.1% per year. Here we have the NDVI maps for 2010 and 2020. So here, the red areas here signify the dense vegetation. So as you can see, it is mostly visible because most of the mangroves of Sibuyan for both 2010 and 2020 are dense, classified as dense. But here you can see that there is a visible expansion. So later I will show you a closer picture to see the expansion. All right. So here we have the 2010 map. And as I have mentioned before, the Sibuyan Island is composed of 3 municipalities, Magdiwang, Kahidiocan, and San Fernando. So this tile here, A1, this is in the part of Magdiwang Municipality. This A2 and A3 is in Kahidiocan, and A4 is in San Fernando. And if we will compare the 2010 map with the 2020 map, so we can see that there is a visible expansion of the red areas, which are the dense vegetation. So this is 2010, 2020. So what mangrove species compose these mangrove community? So basically, they are dominated by Abyssenia, Soneracia, and Rhizopora, which are basically also the most abundant genera of mangrove species, I would say, in the Philippines. And about 14 species of mangroves are recorded in the province. And here in the photos are some examples of the dominant mangrove species in Sibuyan. So we have here Abyssenia marina, Soneracia alba. So basically, mangrove species are classified or identified based on their leaves and fruits, and also based on their root system. So here you can see the specialized root systems of Abyssenia marina and Soneracia alba. Does anyone know how this is called? So they are called pneumatophores, which are basically acting like snorkels for breathing during inundation periods when the water is high enough. And here, in the last picture, so we have here a Rhizopora species. So it could be Mocranata or Apiculata. And as you can see, it is characterized by its elongated fruits and its prop roots for structural support. All right, so to conclude, so this study demonstrates the use of Landsat images in detecting and monitoring changes in mangrove cover on an environmentally critical island like Sibuyan. And as we have seen, there has been an increase in the mangrove cover and vegetation density within the 10-year period. And when we looked at the possible drivers of change, we have found that there are several planting activities and protection measures that were implemented in the area from 2010 to 2020. And so the increase could have been potentially driven by assisted regeneration through active planting and natural recovery through stringent protection measures. And in fact, these planting activities and protected areas were concentrated in Magdiwang and Kahidiocan, where, as we have seen, significant increases in the mangrove cover were observed. And given these findings, so these are our recommendations to maintain and further improve the mangrove cover of Sibuyan Island. So first is to ensure the continuity of the programs that are already in place, particularly rehabilitation and protection programs. And we also recommend to prioritize the use of Abyssenia and Soneracia species for seafront rehabilitation projects, because these areas are exposed to strong tidal waves, and Abyssenia and Soneracia are naturally found in these areas, so they are adapted to these conditions. Unlike Rhizophora, which is kind of growing landward after Abyssenia and Soneracia. So there is like a spatial zoning of these mangrove species. And also, since mangroves naturally grow at or above the mean sea level, we recommend that they are planted in the middle and upper intertidal zones, and they should not be less than 50 centimeters. So ideally, they should be 0.5 to 1 meters in height. And also, although this will involve further studies, but the incorporation of green-gray engineering in addition to planting can be considered. And also, it's important to create long-term sustainable livelihoods for coastal dwellers. And one of the potentials could be nature-based tourism, since the area is also rich not only in mangroves but also in marine resources. And it's important as well to integrate the efforts of organizations and local communities toward mangrove protection and to boost networks and strengthen financial support for research and extension on the island. All right, so these are my references, my acknowledgement to my co-authors and the DAAD and University of the Philippines, Los Baños. Thank you very much.
We have no time for the questions, so please keep them for the discussion later. Now I invite Magdalena Maichrzak, who will tell us about the possibility of using Sentinel-1 radar in vegetation dynamics characterization.
So, hi everyone. Thank you for the introduction. My name is Magdalena, and today I'm going to show you how early spring vegetation dynamics in deciduous forest can be relieved by radar observations. So, first of all, spring phenology of deciduous forest is a sequential greening of layers. There are different layers in the forest, and after the dormant season of winter, with some warmer days and more sunlight, light reaches the bottom part of the forest, and therefore the forest floor can start developing. Then eventually with time, other layers will develop, but it's a sequence of events. It doesn't happen all at once. And this shift from winter dormancy to active growth is accompanied by beginning of photosynthetic activity and an accelerated increase in carbon uptake. Also, this development of early spring is a food source for a lot of animals. Therefore, this has a lot of ecological and climatic impact. And those changes of no leaves into open buds can be very rapid. And usually the observations can be conducted in two ways: from the ground, by people going to the forest and observing the development, but also using the satellites. And about the satellites, we can distinguish optical and radar data, and there are some significant differences between them that will influence what the satellite can really see. So before you could also hear the presentation that were used optical data, and basically optical satellite needs sun as a source of energy to make the surface bright and to capture the image, but also it's usually blocked by the clouds. So for the early spring development of forests, especially in Poland or region of more north, there is a lot of cloud cover during the spring, and therefore a lot of images are basically clouds. While radar can penetrate through the clouds and can see the surface without being blocked. And we can say that optical tells what the surface looks like, and radar can tell how is it structured. So radar can have different wavelength, and this, like, the logic is pretty simple. The length of the radar beam will tell what the satellites see. So for example, if the length is small, like 3 or 5 centimeters, it will usually interact with small parts of the forest, for example, branches or leaves. But if the length of the radar beam is longer, it will penetrate through the forest. And for example, with very long band, it can even penetrate through the ground. And so the methodology of this study is the use of ground observation and integration of radar. And the goal is to relate those ground observed phenology and structure to radar-derived backscatter and coherence to understand and monitor vegetation dynamics. And let me just also explain what are those backscatter and coherence are. So basically, when the radar emits a pulse, the pulse reaches the ground, it interacts with whatever there is, and then it goes back. And this is influenced by the structure of the surface, the moisture, and for example, if the surface is very moisture or even water, the signal will basically be bounced away, and a very small amount of the energy will go back to the radar. But for example, if there is a lot of vegetation, it will interact with the leaves and different parts of the forest, and more part of the radar signal will go back. And there's another thing like coherence. It's a little bit different. For coherence measure, we need at least 2 different images, because coherence basically tells how similar 2 different images are. It's very precise. Usually it's used to monitor volcanic activity or landslide. It can tell very precisely centimeter differences of the ground. And usually vegetation cover is considered as noise for this kind of measurements. And in this study, we try to measure this noise and make use for it for the forest observations. And basically coherence ranges from 0 to 1, and 1 will mean identical, but very rarely basically it never happens, because there's always some differences, even if the atmosphere part that also influences a bit the results. And 0 will mean no similarity at all. So the study area for this project is located in Czerniejewo Forest District, and some polygons based on Forest Data Bank of Poland were chosen, with fully covered by Betula pendula tree, birch tree. And the results from SALCOM. So just to remind you that I told you that different waves interact with different sizes of the forest. So this SALCOM has 24 centimeter wavelength, which will mean that it is quite not sensitive to small changes. And there is this big drop, as you can see, in the middle of February, and this is because of the snowfall. Radar is very sensitive to a lot of moisture changes, but beside that, the signal is pretty stable. When it comes to different satellite wavelength, Sentinel-1, that has much smaller wavelength, you can see that the signal is much more fluctuant. And if I show you how does it look compared with precipitation, you can see that, maybe I can actually use this. Okay, you can see that those fluctuations are a lot of times a reason of precipitation. So backscatter is very influenced by moisture. Therefore, we also use the coherence. So again, coherence will mean that low coherence means no similarity between 2 different images. High coherence means a lot of similarity. And the lowest coherence results are happening here in the beginning of April and middle of April. And now when I will show you the images again from the ground observations, you can see that the biggest difference in the canopy happened in the middle of April. And also about the forest floor started developing earlier, and those images of 27th of March showed that there was already vegetation there before. So here I have also a graph showing optical against those radar data, and in general for the period of January until June, there was 45 images available. However, only 14 were useful because of the cloud cover. And there is also a rise of this NDVI value, which is, I will remind you, it's a green index showing how green the area is. So it shows that the area is greening up, but it can be it's not sensitive enough to show the greening of the forest layer, especially if there is a lot of cloud cover during the observation period. So the main conclusions are that this interferometric coherence can capture those early spring changes in the forest, and the temporal decorrelation is linked to the vegetation development. But compared to the coherence, the backscatter is more affected by precipitation, and the optical NDVI captures the main phase of canopy greening, but it can fail to detect those early structural changes. Okay, that's all. Thank you.
Thank you. Now I would like to invite Magdalena Kurłowicz, who will tell us what can we learn from tree rings.
Good morning. My name is Magdalena Kurłowicz, and I am student of landscape architecture at the Wrocław University of Environmental and Life Sciences. This project has done together with my supervisor, Monika Ziemiańska, professor, and she guided me from very beginning. Also, research is part of a project named Fragile Heritage, as a part of Interreg Poland Saxony project, co-financed by Interreg by European Union. And the title of my talk is Tree Rings as Climate Archives. And in next 15 minutes, I would like to show you what Scots pine in Lower Silesian forest can tell us about climate. But probably even more, I would like to show you the whole process, because for me it was most interesting. Let me start with the aim of the study. The first one, to build the site chronologies for 6 selected pine stands. Also, we wanted to assess how Scots pine respond to monthly temperature and precipitation, but we wanted to do that for total ring width, as it usually is done, but also for early wood and late wood. Third goal was to identify which months of the year drive pine grow, and fourth to compare climate grow response between managed forest and strict nature reserve. And maybe you don't know it yet, but annual grow rings are so interesting because in the small layer of the wood, tree records experience during the year, and there are many factors like weather, like drought experience, or human pressure like pollution. And my job was to separate those signals from each other. A little bit of time scope and climate data. The field work was carried out in last summer. For climate data, I took monthly mean air temperature and monthly precipitation in totals for period 1951 and 2024. And one more thing worth of mentioning is that growth year is not the same as calendar year. It's not from June to December, but it starts in previous October and it ends in September. And this window led us to capture the delayed influence of on tree. Here is where I worked. It was in Lower Silesian Forest. It's one of the largest complex in Central Europe. It's located in southwestern Poland, near to German border, and it's not well studied, which is perfect. Its forests there are dominated by Scots pine, and its most important forest-forming species in Poland, both ways, ecologically and economically. But for me, there was one more reason to study Scots pine, because it's climatically sensitive. And it records the conditions of growing season into every tree ring, and in this way it's climate archive. Here you can see where exactly I worked. I had 6 study plots, and 3 of my first research area were in strict reserve, protected forest called Przymgiełkowe Moczary, and 3 other 3 forest research areas are in normal managed forest. And let me walk you through the whole process. At first, we have to choose at least 21 trees per plot. And it's tricky because not every tree in forest is the same. Foresters describe this like as craft classes. We have 5 of them. And for our research, we took only first and second class because we had to exclude third to 5. They grow under the pressure of their neighborhood, and they don't have perfect, clean signal. Also, we had to choose healthy trees with no visible defects. Then we use tool named Pressler increment borer. It's 5 millimeters wide. We pull it into the trunk, turn it into the trunk, and pull out the thin core. We have to protect every core, label it, and transport to the laboratory, where, of course, work is continuous. Sample preparation goes first. Samples are glued into wooden holders, sanded and polished for many hours to make tree rings as visible as we can. Then we go through measurement. We take high-resolution scans of our polished cores. We measure it in Kórnik program with accuracy 0.01 millimeters. And usually we measure only total ring width, but I went step 1 further, and I measured early wood and late wood separately. Then we make synchronization for every plot and check it in Kofecha program to check if everything is matching. If something is questionable, we go back to sample, check everything, and sometimes we have to remeasure sample. Then we have chronology. We build it in Arston program. We have tree chronology, raw, standardized, and residual, but it's for next slide. And also we check EPS index. It says how representative our plot can be. And also to the heart of my study, climate grow analysis. I did it in RStudio with package TreeClim, and climate data was in 12-month window, as I mentioned before. It's dependent variability, and chronology, residual chronology was dependent variability. Sorry. And results. I built the 6 chronologies. for every plot. We have a different line, and every chronology is in 3 components of wood, total ring width, early wood and late wood. The oldest series have 153 years old, and it's from 1871. And we have to build residual chronology because raw chronology, raw measurement in millimeters, have trends like age trend, and it have nothing to do with climate. It's obvious that young trees grow wider and old trees grow narrower because the same wood spread all over larger circumference. And the standardization I made removes that trend. Here is an example of one forest research area result. And bars are correlation coefficients. It made by a bootstrap method. It program made 1000 of calculates, and in slightly different with slightly different data. And if the coefficients stay strong, we can say the correlation is statistically significant, and it's the bolded, vibrant blue bars. Line is response function, and also I checked everything in Statistica, and it's the nodes with picture you can see here. Also I checked R squared, and its total percent of variability of growth explained by climate and months together. It means, on example, for late wood rainfall, we have R squared 42%. It means 32% of variability of year to grow could be explained by rainfall. And key findings. First one is across all over 6 plots we observed similar trends, which is good because they talk to each other. Maybe I can say that. We know that woods drive early wood grow the most, and it's temperature, especially in February and March. And it's positive correlation. That means warmer conditions, it's wider early wood. And R squared is 12 to 35, which is not that much. And late wood, what late wood drive grow is precipitation. June to July, it's the most important months, and more rainfall, it's wider late wood, and R squared is up to 60%, which is really strong signal. Total ring width is combination of both signals, and its result says that most of scientists measure only total ring width, and they lost a lot of data not separating early wood and late wood. One ring is 3 different signals, climate signals, and also I checked the protection status, and it does not affect the climate growth response that much if it's forests were managed or it was protected. Thank you very much.
Thank you. Thank you. Now I would like to invite Mateusz Muzow, who will tell us more how oak can live or survive drought in Sweden.
And today I would like to present some results from my master's project a study which I conducted at Southern Swedish Forest Research Center at Swedish University of Agricultural Sciences. So I would like to start with some background. Why oak? Why drought? At Articulate, oak is a species very important from economical and ecological perspectives, and many European countries. It is managed for high quality wood. It supports biodiversity. I found a study which showed that it supports around 2,300 species, different species. However, in Sweden, oak has faced a decline. The abundance has declined by around 80% due to different historical and management changes. Mostly because it has been replaced with agriculture or in forest by pine and spruce, which are fast-growing and very productive species. However, pine and spruce may face very hard time in the future under the upcoming climate or ongoing climate change, and oak may have a potential to even expand its range northward under the climate change. And although oak has a capacity to buffer the negative effects of water limitation, and it may expand its distribution range, climate change is increasing the frequency and intensity of droughts. So my question was, what drought intensity can lead to oak growth decline? And based on that, I formulated following objectives. So in my study, I wanted to quantify oak growth responses to drought. So how the volume growth changes under drought conditions. And I was interested mostly in volume, because volume is what matters from the perspective of practical forestry. I also wanted to identify the drought intensity at which oak starts to decline. And finally, I wanted to see, when there was a growth decline, I wanted to see how long the growth recovery is after the droughts. And I formulated following hypotheses. So the first one was that oak radial growth is primarily limited by drought rather than temperature alone. And the second, the most important one, is that oak volume increment declines only under severe droughts. And the last one, that severe droughts result in prolonged growth recovery periods. I conducted my study in southern Sweden. I was looking for oak-dominated stands, which were at least 65 years old, to ensure that the growth chronologies included multiple drought events. I ended up with 5 sites. 4 of them were located in Tonnersjöheden experimental forest. And even though they were relatively close to each other, because all of them were in the radius of around 10 kilometers, they represented different age classes, different management history, different density, different soil properties, so they were not the same. And one site in the very south of Sweden in Skarhult. And that's how the stands looked. Even though they looked quite similar, site number 1 was 140 years old, and site number 5 was 68 years old. As I mentioned before, I used stem analysis technique. So I collected discs from trees, and I collected discs from every 2 meters along the stem. So stem analysis technique is a destructive method, but it provides very detailed growth information, and I was able to reconstruct the annual volume increment. When I was in the field, I looked at the trees. I looked only at the trees with well-developed crowns, with no visible damage, no double tops, nothing what could affect the climate-growth relationship. So I ended up with 26 trees. As I mentioned before, stem analysis is very labor-intense, so I had to limit the number of the trees only to 26. I collected 263 discs, plus I collected breast height discs just to conduct one of the analysis. This is a tree before and after sampling. And now I would like to say a few words how I did the analysis. I started the analysis with quantifying the drought severity. And to quantify drought severity, I used standardized precipitation evapotranspiration index because it accounts for both precipitation and atmospheric evaporative demand, which is temperature driven. And I calculated SPEI at multiple time scales. However, I selected SPEI 3, so SPEI calculated over a 3-month period because it showed the most significant relationship with oak growth. And to answer my first hypothesis, I conducted response function analysis. So I used the breast height growth chronologies, so only the tree ring width, which was detrended, and I related it to SPEI and air temperature. I was mostly interested in SPEI, so in drought, but I also wanted to see how air temperature can affect the growth. And it allowed me to assess the influence of both climate variables on the growth alone, so I could separate them. And in the end, I identified a month when SPEI had strongest influence on oak growth, and I used it for the subsequent analysis. And as I mentioned before, I used the stem analysis technique to reconstruct the volume, annual volume increment. And stem analysis provides very detailed growth information from multiple heights, and it accounts for the variation in radial growth along the stem. And it's very tricky to reconstruct the annual volume increment based only on historical diameters. And I had to quantify the growth responses to drought. And to do that, I divided observed growth, so observed annual volume increment, by the projected. And to obtain the growth projection, I used flexible polynomial spline, which was fitted to the growth chronology before the drought. And it resulted in the growth projection. So the red dashed line is a growth projection, and the blue line is the fitted spline. And to answer my second hypothesis, I conducted threshold analysis, and I conducted this analysis to identify the drought intensity at which oak growth starts to decline. And to do that, I selected 10 years with lowest SPEI in June. And even though some of the years were not actually drought years, but it provided me a gradient of drought conditions from very mild or non-drought conditions to extreme droughts. And then I fitted polynomial regression to the growth responses. in those years, and it resulted in the threshold. And finally, to answer the last hypothesis, I conducted a so-called recovery analysis. So I used the growth projections, and I defined the recovery year as a year when the observed growth, so the dots reached or exceeded the projection, so red dashed line, and that was a recovery year. And this analysis was conducted only for the 3 driest years in the past 50 years. Now I would like to present some of the main findings in my study. So now I would like to answer the first hypothesis. So I assumed that growth will be primarily limited by drought rather than temperature alone, and that was true. Here we are interested only in the solid red lines, which indicate most significant coefficients for the climate growth relationship. And most of the red solid lines were in June. That's why I selected June as the month which I was interested in later. I was a little bit surprised that I will observe any of the temperature effects, but yes, in most study there are barely any temperature effects. And my second hypothesis was that oak volume declines only under severe droughts. And here we can see the threshold that I identified. So the dots are the growth responses, and here we have the drought intensity. So the growth declines with a SPEI of around -2, which is very, very severe. And these conditions happened only 2 times in the past 50 years at the analyzed sites. And the growth decline was up to 20%. So usually there was no growth decline at all. And my final analysis was the duration of the growth recovery, and here I haven't observed many declines. In most trees there was no decline at all. So the first bar, it was like 60% of the trees, and for the most severe drought, which was 1992. Many trees never recovered, so the recovery exceeded 10 years. And now some conclusions. So SPEI in the spring and early summer, particularly in June, had a significant relationship with oak growth. And oak showed relatively high resistance to water limitation. It tolerated moderate and severe droughts. The volume growth declined only under very extreme drought conditions. And the decline was around 20%, up to 20%. Many trees did not exhibit growth decline at all, even under very severe or extreme drought events. However, we need to remember that I had only 26 trees because it was very time-consuming to process the samples, and it is some kind of an indication. And in further studies, I would include more sites from drier parts of Sweden and different ages or different densities. And thank you very much. That was all.
Thank you, Mateusz. Now I would like to invite Jagoda Pękala. and we will have another lecture about tree rings.
Okay, it's okay. Yes, yes. Thank you so much for the introduction. My name is Jagoda Pękala, and I am the master student in University of Environmental and Science. And with my supervisor, Professor Monika Ziemiańska, would like to talk more about the pointer years analysis in tree chronologies of Scots pine, Norway spruce, and pedunculate oak from the Lower Silesian forest. And as the beginning, I'd like to introduce some main aims. The first, an attempt to analyze the radial growth patterns of three studied species. Second, with synchronization, cross-dating of tree series of three studied species, the Pinus, Pita, and Quercus. The third, the determination of pointer years of the studied species. And the fourth, an attempt to explain pointer years by identifying the environmental and climatic conditions prevailing during positive and negative growth years. And the time frame, our fieldwork was in last summer, in early July and in late August. We collected materials and prepared for measurement in September 2025, and we measurements and analyzes the research material commenced in October 2025. And I can also say that the tree has different type of growth year. It started from the previous of October in previous year and ends it's September of the current year. So it could be the different instead of the year. And why I chose the forest, the Lower Silesian forest. It's a very largest contiguous forest complex in Central Europe, and we also chose the unique sampling location, and it was selected in the area of the former village in Prędocice, and the history is date back to early 15th century. And why the three type of this species? The Scots pine represent the dominant forest forming in our country. The second species included in the study was Norway spruce because it was located in the early medieval archaeological complex of Białogórze, as well as its position near the ecological limits of its habit range. And I also chose the Quercus robur because on this localization, it was the very interpreted as the relic of the former settlement structure, and the material was in a very good condition. Maybe some about the localization. Our study plots were conducted in the southwestern side of Bory Dolnośląskie, and we chose the two forest districts, Ruszów and Pieńsk, and it also every sample, every area has their own code, so we can find on the Forest Bank of Data website in Poland, and every site has different types of habitats. Something about the sampling. Sampling was conducted on days without precipitation, and we also used the craft classification system to visual tree assessment and tree age and percentage contribution of the target species within each stand component were taken into account. And we sampled at least of 20 samples per site. And we used the Pressler's increment borer to increment the course. The another step was pasting. The samples had the form of cylindrical cores and approximately 5 millimeters in diameter and about 30 centimeters, sometimes 40 centimeters in length. Then we prevent during transport. The cores were first secured with the masking tape, and then we pasted them into using wood glue onto the wooden holders. And after that, we carefully sanded and scanned it using Epson and Vision scanners. And it's important to sand it very clearly because we can have the most visible site on this in the program. And after that, we measure it using the co-recorder and cross-dating and synchronize it with using Dendro program. And our reference year was 2024 because 2025 year was an incomplete year for the tree growth year. And the first results, we can see that we can see the different type of data, including, for example, the highest increment, highest maximum ring with increment for especially Quercus robur, or we can see the express population signal and different type of datas. And on this slide, I represent the raw chronology of studied species, and they exhibit similar and closely aligned trends. The growth responses are coherent across site, as if they were talking to each other and forming synchronous patterns that are evident in the upward and downward trajectories over time. You can see the sign lines that are typically shows the lower and the higher signal. And as like in topic of my presentation, the pointer years. The pointer years is an important element of dendrochronological analysis. They reflect synchronous growth responses of trees to extreme climatic condition. We use the threshold 90% to have a strong signal. And when at least 90% of the trees showed an increase in ring width compared to the previous year, it's called the positive year. And when at least 90% of trees showed a decrease in ring width compared to the previous year, it's called negative year. And in total, the 44 pointer years are indicated, including the 25 of negative years and 19 of positive years. And to highlight different shades of green and the red were used to highlight individual years in which at least 3 study plots showed a consistent growth response signal. And also the analysis of negative pointer years over the period 1951 to 2024, identified 6 years in which at least 3 sampling sites exhibited, as like said before. And these years were also characterized by anomalous drought conditions, like severe droughts or deep or extreme. And we can also say that the positive pointer, we can also, I'm sorry, I can start from the beginning. The analysis of pointer years was conducted based on the thermal and pluvial conditions. And this black line represented the average mean monthly temperatures from the period 1951 to 2024. And we can say that the positive years were formed when temperature was, for example, lower in January or February, November previous, or it was higher, especially for Norway sprouse. And the negative years was formed when the temperature was higher, especially in this site. And for the precipitation, we have also the black line represent the average precipitation in the period 1955 to 2024. And we can also say that the positive years were formed when the precipitation is higher in March, in June and July, and the negative pointer years were formed when the precipitation was lower, especially in February or in timeline from April to August. And we can interpret these graphs so you can interpret these graphs as long as you need. And for the conclusions, in total, 123 increment cores were collected, and over than 12,000 annual growth rings were analyzed and measured. The mean values of radial growth were calculated for the study species, the Scots pine, Norway spruce, and pedunculate oak. 3-ring series were carefully and systematically cross-dated, achieving satisfactory values of standard dendrochronological parameters. The site chronologies were developed for the analyzed species, covering the following periods: the longer in Pinus sylvestris from 1845, and a total of 45 pointer years were identified. An attempt was made to interpret the meteorological conditions, temperature and precipitation prevailing during both positive and negative pointer years. And I would like to also thank you for the opportunity to take part in the project Fragile Heritage in cooperation with the Interreg Poland and Saxony program. And thank you for my presentation.
Thank you. Thank you all the speakers for the perfect timing, and I would like to start the discussion. Maybe we can start from the beginning, and I think we will have approximately 5 minutes per speaker to ask the question, so please.
First of all, congratulations for the presentation. Very nice study. I just have one curiosity because maybe I didn't get. In comparison to what did you calculate the accuracy metrics for the study?
Can you repeat the question?
In comparison to what database, which was your control data for calculating the accuracy metric, the kappa, etc.?
Okay. So we use ground truth points using Google Earth, the Google Earth engine.
Okay.
Yeah.
Okay, thank you.
Morning. Congratulations in your presentation. My question is, you show as a result that the dense forest is growing more than the other two, the spare and the bear, I think.
Yeah.
And I saw in the graphic that the dense is replacing the spare. Do you have the same species in the spare and in the dense, or different species and you're facing like something bad?
All right. So that's an interesting question. Thank you for raising that. Actually, the study focused more on the remote sensing side. So we are yet to verify on the ground whether those species were the same or not. But yeah.
Any more question? I have a question about the level. You have presented that on the one side of the island you had lower cover change compared to the other side of the island.
Yes, yeah.
So what's the reason? The species or the environmental change or what different regulation?
Okay, so when we looked at for example, the potential drivers of change, we have found that more of the what do you call this? Of the planting programs and protection measures were concentrated in Magdiwang and Kahidiokan. And so we cannot like conclude totally, but I think that is one of the possible reasons why. Yeah.
Okay, thank you. Do you have question still? No, thank you.
All right, thank you.
So I would like to invite Magdalena Majszak.
No, first of all, congratulations, because explaining synthetic aperture radar remote sensing to an audience which is not based on remote sensing is very complex, and I think that you did it very well. Apart from this, the question, which was the resolution of the L-band product, the SAOCOM that you used for comparison with the C-band?
As I remember correctly, I think it's 23 centimeters.
Not the wavelength, the resolution, the pixel resolution.
Okay. Oh, that's a good question. It has like the pixel size is lower than Sentinel.
Is it even lower?
Yes, it's lower, but the resolution time resolution is higher though. So I think it's maybe 11 days. I would have to check it more.
No, because
precisely.
I'm thinking that, however, maybe with an L band of better resolution, like for example ALOS, you could get better result. Because for me, combining both the C and L band for the study is probably, you can get even better result, just for a future trial, okay?
Thank you so much. Also, for this year, I applied to receive the X-band data with 3 centimeters, and I'm quite curious how it will look, although it might be very noisy because of the band, but...
Thank you very much.
Thank you.
Some other question? I was wondering if this method is used to select area with that crown of trees in the forest.
It can be.
It can be.
It's used to detect, for example, the deforestation areas a lot, but it could be since it's very sensitive to moisture changes, and the dead trees are much less moisture.
Okay.
It would be.
Because I think it would be faster to describe from the above, I think, because on the below ground we sometimes...
But also for this, the optical data and DVI values are useful a lot. It depends, like the other advantage is that it can penetrate the cloud, so you have more data within the year. But the optical is usually easier to work with and is more, like the results are more appearing, I would say, like they're easier to analyze.
Okay, and is there a possibility to describe or differentiate different species because they will, I don't know, accumulate more light, or I don't know? I'm not...
That's a good idea and good question. So, for example, if the species would have, it depends. If you would like to identify exactly what's the species there, that would we would have to have a lot of data to learn the model. But for example, if you would like to say that this forest has different structures, so it means different layer, different species on different layer, that's more easy to do.
Okay. Because I'm looking for the trees growing around the Poland by using Google map and the satellite. Sometimes it's work.
So for
not,
this,
so.
it's much more. You can do it in the automatic way. You don't need to do it yourself. So it's much more fast. And
yeah. Thank you. Thank you. Some other question? If not, thank you.
Thank you.
So I would like to invite Magdalena Kurłowicz. Maybe we can put. Do you have question? Just to keep it to remind you. So I have a question about the tree rings. If there is a possibility that when you compare reserve forest, protected one, with the managed one, you will see the difference in tree rings growth related to environmental of soil, let's say. Have you checked it or somebody was checking it? Because in the protected area you will have different condition of soil, I think, than compared to managed one.
Well, can I? While checking, choosing the forest research area, I was looking for as comparable site conditions as it's possible, and I have comparable habitats, one in managed forest, one in protected. And I didn't see the difference between the grow.
How long this strict reserve was protected when it was established?
It have long protected history, because it's after war, Second World War. If I remember well, there stopped reserve status, but it came back in 2012. Okay. We can say it's questionable because it wasn't reserve for a few decades, but it wasn't managed. The forester says it's not possible to manage this, so not in the paper. It was protected, but it wasn't managed.
Okay, thank you. Some other question?
Oh, sorry.
If not, no.
Thank you.
Now I would like to invite Mateusz. Do you have a question related with extreme droughts?
Good morning. Congratulations. You mentioned at the beginning that you had different environments for your study, and then you have the results showing that some trees didn't decline and others decline. Do you have some environment information, like the best conditions that they declined and they not decline?
Not really, because some of the trees within the same sites declined, and some other trees did not. So it was very difficult to predict that.
Mateusz, my question is, do you know the history of oak forest in Sweden? I'm asking about the early stage of growth of the trees and the source of regeneration. What was used? It was naturally regenerated by acorns, or in Sweden they use seedlings from nurseries, because it's connected with the forming the top root, and this factor is crucial for the drought response after.
So at most of my sites, the trees were planted because the sites are experiments, long-term experiments, and the history is well known. And there was just one site where the forest was naturally regenerated. But I haven't really noticed any differences between those sites.
Congratulations on your presentation. You mentioned about coring 26 cores, right?
Sorry?
26 samples.
26 samples,
yeah. You mentioned about that. So did you exclude some because they have low correlation, or you included everything?
No. I had 26 trees, and from each tree I collected between 9 to 13 samples because I sampled the whole stem, not only breast height.
Okay.
Breast height was an addition just to verify the climate growth relationship and to identify which month is the most important for oak growth.
Right. So how representative do you think 26 samples were?
They are not really representative. I mean, it's some kind of indication because I wouldn't draw any strong conclusions from 26 trees. And one of the parts of my study was combining these results with larger data sets. However, it is very difficult to reconstruct the volume increment, annual volume increment based on DBH. So I'm still working on that, and we are looking at the methods, and we'll see. Hopefully, I will have more data and more results later.
Right. Maybe I missed it. Did you do the EPS probably? The
Sorry?
express population signal? Did you do that?
No.
Okay. Thank you.
Yes, I also have a question about history. So in Estonia, oak forests are really rare these days, but after the last ice age, some 7,000 to 5,000 years ago, they were rather common there, as were all the other broadleaves. And that was a period of time that was warmer than now and more moist, and it is called the Atlantic climate period of Estonia. And since then, all the broadleaves have declined there, and a lot of them have been replaced by conifers. And I'll ask, are you aware of anything similar happening in Sweden, and how common are the oak forests these days in Sweden? Do they often naturally regenerate, or are they mostly planted?
These days, oak forests are not common at all. The abundance of oak has reduced by around 80% over the past centuries. And I think it's mostly related to the industry, what industry needs. And industry needs pine and spruce because they are easier to grow, easier to process, and Sweden is not like a country which is producing for high quality wood. They produce for volume. They manage forests for volume. So oak is not the best species to manage for volume.
Hello. How big is area of your study? Like in hectares or something? You say it's only 26 trees and that's enough. I took 35 and it's enough on 3,000 hectares. How big is yours study area?
So I sampled the trees within long-term forest experiments, so they are relatively small. Each site is around 1 hectare.
Then it's 26 hectares per all.
No, I sampled 5 trees per site, or 6.
Okay.
So 5 trees per hectare.
Thank you. Or there are other questions? No? Okay, thank you. Because we have 5 minutes, let's say, and we can ask Jagoda about her lecture. So, to remind you, the topic, do you have a question related with this? No?
If no, it's okay.
I have a question about this 1957. It was the first year of, it was marked as a positive year. What happened at this time in Poland? It was raining or what? Because
It was in, I think, the droughts. Maybe I can.
I was wondering,
Okay.
because you showed that there was data related with the growth since 1980 something, and now we have the, let's say, first, or it was much more positive year before this 57, or it was just the time starting?
It was a positive year because the precipitation in 1956 was higher, and the trees started to do a long growths, ring growth. So the 1957 is the positive year for these trees.
So before it was also drought years.
Yes.
So it's not okay. Because I was wondering if it's the first one or you had the previous also positive years, but okay, thank you.
We can calculate the precipitation and try to understand the growth season of year and try to understand why the shrinks were the longer than the earlier or something like that.
So most of them, as according to the table, but I think it's also related to your data. So Scots have much more positive years than compared to other oak or spruce, or it's just the examples.
Yes, we can say that the Scots pine plots are quite similar to each other, and then after the research and analysis, I can also say that the quercus have the longest, the negative years instead of these plots, and yeah.
Okay. So according to this, your data, Scots should grow better in our environment, let's say.
Yes.
Okay, thank you. Do you have a question? If not, thank you all of the speakers. Thank you for the presentation of your data and the perfect timing. And I invite you for the coffee break. Thank you.
Let's start. Good morning, dear colleagues. Welcome to Room A, Session 3 of Second International Conference, Research and Practice in Forest Ecology, organized by Institute of Dendrology of Polish Academy of Science. My name is Olena Blinkova. I will share this session. Today we have 4 presentations covering different but connecting aspects of forest ecology, understory regeneration, vegetation changes, urban tree pathogens, and fungal diversity. Each speaker has 15 minutes, and please keep your question for final discussion. So let us begin, and I would invite first speaker. Our first speaker is Zofia Sabocińska, who will talk about post-agricultural forest regeneration using the Forest Maturity Index. This is a very interesting presentation from my point of view for understory regeneration, especially in landscape where forests are developing on abundant agricultural land. So, Zofia, please go ahead.
Hello, everyone. I'm Zofia Sabocińska from the Institute of Geography and Special Organization, Polish Academy of Sciences, and I will present the topic from the final part of my doctoral thesis concerning modeling post-agricultural forest regeneration using the Forest Maturity Index. First, I would like to introduce you my research area, which are Polish Eastern Carpathians, which were interesting for modeling post-agricultural forest regeneration because of population displacement in 1940s, which led to large-scale agricultural land abandonment. And it caused spontaneous and partly forestry-assisted regeneration. And as a result, extensive post-agricultural forest area developed here, expanding the already huge area of predominantly well-preserved ancient forest. To perform the research from the topic of today's presentation, we developed first a map of post-agricultural and ancient forest in this area on the basis of the aerial photographs from 1944, satellite images from 1960s and 2020s. And there are 96,000 hectares of ancient forest and 43,000 hectares of post-agricultural forest, which were created just after the displacement. And I performed field research on 294 research plots, and I took phytosociological relevance, hemispherical photographs, and soil samples. And you can see the example of my field research plots in recent forest and in ancient forest. As you can see, recent forest here is our post-agricultural forest, and ancient forest in this meaning are forests with long continuous habitat history. So it's not about naturalness or really high age of trees. And as you can see, the age of trees can be quite similar, but the difference is, the bigger difference is in understory species composition. Our aim was to evaluate understory regeneration, but first we checked whether the presence of forest-forming tree species was comparable. And only pine, which was introduced here as a pioneer species, was not consistent with wasn't consistent with a natural habitat, showed statistically significant difference in coverage. Nevertheless, it is worth noting that it never reached 100% cover in our study plots. And according to the results of the trended correspondence analysis and canonical correspondence analysis, the primary factor differentiating the research plots was former land use. So whether the plots were the research plot was in recent forests or ancient forests. And then on the basis of comparison of data from phytosociological database, I created a list of ancient forest plant species for the region. But this is not topic of today's presentation, but that was one of the steps to collect the final part of my research. So I'm not going to tell about it anymore. But there were 15 species occurring significantly more often in ancient forest than in recent forest, and also 9 species occurring significantly more often in recent forest than in ancient forest. And getting to the point, I measured forest regeneration on each research plot by forest maturity index. And I divided number of ancient forest species in each of forest plot by number of ancient forest species indicated for our study area. And these are the results. This is the map with each of my research plots. And the circles on the map indicate areas in post-agricultural forest, and triangles indicate areas located in ancient forest. And the understory regeneration rate measured by forest maturity index for this 70 years old Carpathian recent forest varies from 0 to 0.54. But it's worth to see that FMI value, for example, 0.5, does not translate directly to 50% of regeneration, because the reference value of FMI in ancient forest never reached 1. So just because there were no such situation that on one research plot there were all ancient forest plant species from the list. But this is the comparison of forest maturity index values in all recent forest and ancient forest, and you can see that in comparable environmental conditions, the regeneration rate of post-agricultural forest can be comparable to the median in ancient forest. And to investigate which factors have significant impact on regeneration rate, we counted a multiple regression model where dependent variable was forest maturity index, independent variables were elevation, slope, topographic position index, heat load index, compound topographic index, canopy openness, carbon, nitrogen, sulfur content in the soil, and distance from the ancient forest. And as a coverable was pine cover, because the recent and ancient forest plots differ significantly in terms of pine cover. The results of regression model showed that the most important factor affecting the regeneration of this mountain post-agricultural forest is distance to the ancient forest. This is the source from which ancient forest species can migrate to recent forest. Second important factor was slope. The steeper slope, the regeneration was better, maybe because the deeper influence of agriculture in flat areas, and perhaps the activity of foresters was greater in flat areas. Elevation, the higher elevation, the regeneration was better, because the the higher elevation, the regeneration was worse, not better, because of less favorable climate and poorer soil, for example, and pine cover. The more pine in a tree stand, the regeneration is also slower, worse, or because the pine was introduced here as a pioneer species and is inconsistent with natural habitat. And based on the values of this significant factor, calculated for each 40-meter grid cells across the study area, we modeled level of regeneration. And our results showed that forests with higher FMI values tend to occur in smaller, more fragmented patches compared to areas with poorer regeneration. This is likely because these well-preserved, regenerated sites were often surrounded by ancient forests and were located in less accessible areas, which, for example, limited conversions into large-scale pine plantations. In contrast, the red areas visible on the western side and here on the northern part of the map indicate extensive pine plantations. There were larger patches, and this site were more accessible due to presence of roads and railway lines, which facilitated large-scale land use changes. We can observe that recent forests in the central part of the map near Solina Reservoir showed better regeneration. This region was difficult to access, and after the war there were few wards, and the post-agriculture areas existed as small patches surrounded by ancient forests. To sum up, because of high conservation value attributed to ancient forest plant species and already proved correlation between the presence of ancient forest species and overall forest biodiversity, these results enable the identification of well-regenerated and thus maybe ecologically valuable post-agricultural forests. Having visited this area many times, I observed beautiful and biodiverse post-agricultural forests where natural processes clearly predominate, like on this photo. And if you would like to have more information about this project, I recommend you my publications. First, regarding to the map of post-agriculture and ancient forests. Second, regarding to the list of ancient forest plant species. And the third, regarding to the post-agriculture forest regeneration modeled by Forest Maturity Index. Thank you for attention.
Thank you very much for your excellent and interesting presentation. And I would like to invite our next speaker, is Natalia Kisel, who will present research on vegetation diversity changes in the Dendrology Nature Reserve and the disintegration of Thermophilus oak forest. So Thermophilus oak forest support rare species and highly sensitive to management changes. Well, Natalia, go ahead.
So yeah, hello, thank you for introducing me. My name is Natalia, and I came here from Warsaw, and I would like to tell you a bit about a very rare type of woodland that which are Thermophilus oak woodlands. There are some kind of biodiversity hotspot, you can say, because of the history that made them like this, because they're not natural woodlands. They they are like this because of a traditional management, and such as grazing and thinning cuts. So they developed to be a refugee for rare species because of human action. And in the 90s, scientists observed the danger for this type of forest. And because of the studies, we try to protect these fields by making it a priority habitat in program Natura 2000. And this map shows some of the borders and some of the stand representing the Thermophilus oak woodlands. And as we can see, we have many of them in Poland. But in my study, the area is in the North Poland, which is like the border of this natural woodland type. So to aim is to access the dynamic of diversity in Thermophilus oak forest, like I said, in north part of the range in Europe, and it's also the north part of the range in Poland, of course. So we claim that the Thermophilus oak forest is undergoing a transition towards to an oak hornbeam woodland, and more, that in the reserve flora the proportion of light demanding and grassland species is decreasing, while the share of oak, hornbeam and forest species is increasing. And also that decline in both alpha and beta diversity of vegetation is expected. So now how we make the studies, how we did it. So we choose the study area, which is the Dębowe Góry Nature Reserve. It's almost 100 hectares, and it was designed as a nature reserve in 2001. So it's not that long since it's protected. And I think the important factor is that the nature reserve is located near to the village. So, like I said, the history that causes this type of woodland is because of the human actions, and close to the village is like a fact that we aim that it's important here. So, we have historical data published in 1998 by Professor Sokołowski. And this is the original map from his studies, the location of the plots in which we remake the studies like professor, and the method is relevance. So then when we get the data, we did a cluster analysis by using words method, and also we showed the PCR ordination of phytosociological data. And then we like to show the changes in the habitat preferences, the vegetation beta diversity and alpha diversity. And also we like to show the changes in the forest affinity of the herb layer in the reserve. So now let's go to the results and I want to show you how this reserve vegetation changes. So at this cluster analysis, we see all the relevés, and the green ones are the historical data, and the yellow ones are the current data from 2025. As we can see, there's almost a clear movement from the historical data and the current data. And this was the base for us to identify this type of woods. So we identify the first tree group as Tiliocarpinetum, and we also wanted to show that this Tiliocarpinetum is not the same in every current relieves. So we split them into 3 different subtypes of Tiliocarpinetum, but they're mostly current relieves, and more of almost every historical relief is a type of Thermophilus oak forest, such as Calamagrostia quercetum and Potentilla alba quercetum. So after this identification, we would like to show you how these plots are in the PCOA dissimilarity with Bray Curtis. So we can see now that the movement is also almost clear, that the change is visible. So how does the vegetation changes? Again, as we can see, the index for temperature and light drops, which means that historically the temperature and light were better for rare species that needs it. But now, on the down plots, we can see that there's an increase in moisture and the nutrients index, which is like a consequence for drop of temperature and light. And the other consequence is the change in species composition by using the EU for plant list, that we can tell that every species which is his affinity to forest. So historically, there was more species which are not typical to find in forest, more like to open space, more like mealworks or grazing fields. But now this proportion is changing because we see the increase of typical forest species, and the dark green color is like the forest species specialist. So we see this difference and shift. So here we can see the changes in alpha diversity. As we can see, the number of species of taxus in taxa S index is dropping, while the Shannon and Pillow index are increasing. And here are the changes in beta diversity. We use Bray-Curtis similarity, because in the past program, Bray-Curtis is not a distance, it's a similarity. And we also use Jacquard to show that that beta diversity is increasing, because now the plots are more different than they were in the history. In history, they were more similar. I like the saying that in nature there's no awards or punishments, there are only consequences. And I would like to tell about the consequences of these actions. So, in history, until first half of 20th century, we have grazing and extensive land use in Poland, in this part of Europe. That was the thing that allowed to these Thermophilus oak woodlands to develop. and to be this like refugee for rare and important species. After in the second half of 20th century, there was no permission for grazing and extensive land use because of the laws, because of the changing, and this change has an impact of biodiversity because it happens that because it proved to us the studies that if the forest is more and more nature, then we have the alpha diversity loss. That's what is happening here in the reserve. And now, in 2025, we see that the beta diversity was increasing, and now we have the peak of it. But it's not temporary, because we know from the other studies from the West Poland, such studies like Jakubowska-Gabara, that in this type of woodland, without an active conservation methods, we would have a decline of beta diversity, and later we would only have the type of oak hornbeam forest, and this place would not be a refuge for rare species. And why we can tell it? Because in 1993, the Professor Sokołowski shows that in the flora of the reserve, we can see such important and protected by law in Poland species. And in 2025, we found only the half of them. That's why the increase in beta diversity is not always compared with increase in alpha diversity, and that's why it's only temporary, that now we have a high diversity, but it's going down, and without active protection, this would happen. So, how to protect the specific woodlands, how we can do it, because it's a very important fact, as we have Natura 2000 programs in Europe, and we are we are obligated by law to do it. So, in my opinion, we should find hotspots in these places, like local hotspot, which could be a nature treefold gaps. This is the place where the light conditions are the best, and we should prevent them from growing. And the potential threat in these treefold gaps are the expansion of species like Coreus avellana. So the good practice advice is to cut this Coreus avellana not near to the root collar, but on a certain height, and leave one shoot. Then we prevent this species from growing from the collar root, but growing upper and giving less shadow to the wood floor. Another important factors, which is active conservation methods, like I said, maintaining an open tree canopy and preventing this natural happening canopy gap in the stand from closure. And we can do it by cutting and with help of animals. We can also, at this moment, when we have gaps in the stand, we can start grazing. And another, in my opinion, interesting thing is that we identify a rare type of forest in this part of Poland, which is Acidophilus oak forests. We know this from studies that it's more common in West Poland, and more and more studies shows that the border is increasing and it's changing in the north of Poland. And the studies from 2021 shows that the Vistula River may be the new temporary border for this type of woodland. And the area of the reserve, which is the red dot, is more than 100 kilometers from the Vistula River. So it means that we need more studies in changes of boundary of nature range of woodlands in Poland also. So what happened in the reserve? We see that decrease in the indicator values of temperature and light, and increase of in the indicator of nutrients and moisture. We observe the biotic differentiation of vegetation. We see the increase of beta, which, like I said, is temporary, and it's combined with fluctuation of alpha diversity. We observe the increase of portion of forest-specific species and the progressive decline of important elements like rare and protected plants of Thermophilus oak woodlands, and the presence of Acidophilus oak forest community in the northeastern Poland. Also, the regression of Thermophilus oak forest is accompanied by successional regeneration in the direction of oak hornbeam forest. And the research is limited by not many studies in this area. So to confirm our observation, we would like to study more these fields in the north eastern Poland. And to conclude, despite the changes in climate that should be positive for this type of woodland, we still observe the decline of Thermophilus oak forests, which means that without an active conservation, we are going to lose this biodiversity, these rare refuges for important species protected by law. And as long as we are in European Union and we are part of program Natura 2000, we are obligated to prevent further biodiversity loss. And I think that this type of woodlands are a hotspots for biodiversity and the places that need more attention than they have, especially in the northeastern Poland. And from this place, I would like to say thank you for help of the supervisor, Dr. Piotr Zaniewski, and my dear friends who were helping me in the field. Thank you very much.
Many thanks, Natalia, for your contribution. And next, I would like to invite our next young science. Our next speaker is Joanna Barton, who will speak about phytopathogenic fungi of deciduous trees in Szytnicki Park. This presentation connected. Okay, connected. Oh. This presentation connected urban forestry, biodiversity, and plant pathology. Please.
Yes, hello. My name is Joanna Barton, and I would like to present you the research that I carried out with my student scientific association of plant pathology, Armillaria. at Wrocław University of Environmental and Life Science, and the topic is diversity of tree pathogens in urban ecosystem, and we're focusing on trees in Szczytnicki Park in Wrocław. First, I would like to say a few words about urban forestry itself and the characteristic of the urban environment. The urban forestry is defined as the management of individual street trees as well as city parks and larger woodlands complex within the city areas. And this field faces numerous challenges that are caused by the characteristic of this habitat, such as limited space for roots, for crown growth, and the soil compaction, and the high level of pollutions. Sorry. In urban environments, trees are affected by a lot of stressors that weaken their condition. These include abiotic factors such as restricted growth space, chemical exposure, for example, salts in the winter seasons, and the urban heat island. As well, effects of mechanical damage during the conservation or acts of vandalism. What more, cities are often hotspots for invasive pathogens and pests. This also is caused by the characteristic of this environment, invasive plant pathogens. The cities are working as some kind of entry points for those invasions. The pathogen is introduced very often with exotic plant species that are planted in botanical gardens or parks. The weakened hosts and the elevated temperatures in the cities affect the disease developments, and this leads to these so-called bridgehead effects, where urban environments serve as a starting area for invasion when the pathogens and pests can establish and then spread to more natural environments. And also this process is closely linked with two phenomena that are linked with invasive plant pathogens. First one is spillover. It's when the exotic plant comes with a new pathogen, and this, for example, fungi, then transmit to a native species, and the other one is spillback. And this occurs when like native or common plant pathogen, it infects exotic hosts, often with mild symptoms, and it overwinters on this exotic plant, and then it reinfects native species with increased virulence. The area of study was Szczytnicki Park. It's the largest park and oldest park in Wrocław. It has over 100 hectares, and we can find over 450 species of trees and shrubs in there. Since this was scientific circle research, I focused on smaller area. It's the around 7 hectares part of the park. The whole park is like, is this cursor somewhere here? Oh, it's here. This is the area of the whole Szczytnicki Park, and this is the part that I focused on. The first goal was to inventory and conduct health assessment for trees in this part. I inventored 302 individual trees. And, okay, not this way. Okay. The condition of these trees in this part of the park was assessed as moderate. Many species were infested with mistletoe. Many showed signs of dying branches or of dieback. Many bore visible fungal fruiting bodies, and as well we find symptoms of powdery mildew, for example, on American bird cherry here. And what's more, I collected various leaf spots for further analysis. And here's a quick look at the methodology. The collected plant tissues were sterilized in sodium hypochlorite solution. Then they were plated on PDA medium in Petri dishes and incubated for a week. Later, the representative strains were selected based on the morphology, and they were used for DNA isolation. The DNA was amplified via PCR with specific genetic markers, which were ETS, LSU, and beta-tubulin. Then, after sequencing of those strains, we conducted phylogenetic analysis to ensure the accurate species assignment, and also for specific strains, we conducted artificial infections. They were carried out the way that from the fungi colony, they were cut like small discs. They were put on the asymptomatic leaves. The fungi was left on the leaf for about 24 hours, and then the leaves were kept in a humid chamber for a week. And then under the microscope, I was looking for the structure that this fungi created on the leaves. And I would like to show you some of the most interesting species that we find in this area of the park. Starting with Lotophoma spirae, which is a rather new species because it was first described in 2020 in China and hasn't been described on, I isolated it from the sycamore apple, and it's the first time it was isolated from this genus because first it was isolated on Spirea, yes. What's also interesting about the species, it's first report of this fungi in Europe. And I think it's actually like a fourth report of this fungi at all, since it's rather new taxa. Here you can see the colony of it on the PDA medium and the conidia spores that it's creating under light microscopy. Here is the phylogenetic tree that was created on ETS, LSU, and beta-tubulin markers. You can see that the strain grouped with Lotophoma spirae. For the artificial inoculation, they were carried out on both sycamore apple and also peniculate oak. The choice of oak was prompted by the fact that when I was carrying out single-gen analysis, it pointed that it would be Notophoma quercina. Later, I found out it was different species, but I already conducted the infections, and actually they came out positive as well because the fungi created structures. These little black dots are pycnidia. It's structure where the fungi creates conidia. It looks like this under the microscope, and the conidia came out of this. This is like a bottle kind of shape that it's planted in the leaf tissue. Next interesting species is Spirococcus smithogilvi, which has another name as Gnomnospis. It was actually assessed as Spirococcus this year in March. This one was isolated from leaves of sweet chestnut, and it's actually an emerging pathogen that damages chestnut fruit in the whole world. It has been reported in Europe, but hasn't been reported in Poland yet. And I'm waiting for the sweet chestnut to grow leaves because I'm really excited to carry out infections of this one because I would like to monitor the spread of this disease in the Szczytnicki Park, since the sweet chestnut is pretty common in this specific part of the park. Again, here is the phylogenetic tree, and you can see the strain that's really grouped with this species. Another I think my favorite of those species that we isolated is Tobackia drinia, which was isolated from leaves of Japanese pieris, which is very interesting that Tobackia species is well known as pathogens to all different species of oaks. It's mostly connected to Quercus robur, and it creates pretty similar symptoms on Quercus as it created on pieris. This is also first report of this fungi in Poland, as well the first report of it from pieris, because it usually infects oaks. And here's the morphology on Pidia and the Conidia. And again, the phylogenetic tree, you can see that with 100 of assurance, it grouped with Tobackia drinia. And for the artificial inoculations, I carried out them on both pieris and oak, because I wanted to check if this strain can also infect oak. And while I found the fungal structures on both of those species, the necrosis developed only on pieris. But I'm also planning to carry out inoculation on different species of oaks, because as I find out, every species of oak that this fungi infects can be found in Szczytnicki Park. So I would like to check them all. The next example differs from the others as it concerns more common plant pathogen, Boeremia exigua. We isolated it from the invasive plant American beard cherry, which it hasn't been previously recorded from this host. And I found literature that suggests that this fungi also can infect maples. So I wanted to try if it can create the spillback phenomena, and it did. As you can see, the symptoms of American beard cherry were less severe than those created on maple. Here again is the phylogenetic tree, and the last species I wanted to mention is kind of a honorary mention, because I couldn't carry yet more artificial inoculation because of the infecting the petri dish with Fusarium species, and I'm currently resurrecting the Aptognonomia hystrix. It also was isolated from sycamore maple, and what's interesting, it's like well-distributed maple disease pathogen, and I would like you to conduct some monitoring of spread of this pathogen on maple in Wrocław. And here again is the phylogenetic tree, and you can see it grouped with Aptognomia hystrix. And to conclude, Szczytnicki Park, with its high biodiversity of trees and shrub species, serves as a reservoir for both common pathogen species as well as the emerging one. The moderate height assessment of those trees can be the reason why the pathogens can thrive there. And also we have the positive results of the spillback phenomena, and the genetic markers prove essential for species level assignments of pathogens, because with fungi taxonomy right now it's mostly molecular markers, because morphological description of those species strains is pretty hard because they look very much alike. And yeah, thank you. Oh, thank you now.
Many thanks, Joanna, for sharing your research with us. It was very interesting. And now I would like to invite our final speaker for this session, Martin Garnier, please, who will present research on above and below ground genetic diversity and ectomycorrhizal fungi, and this topic about relationships between genetic diversity in soil and and between sporocarps.
Yes,
Please,
absolutely.
the floor is yours.
Thank you very much. Oh, sorry. After this very good presentation about pathogen fungi, let's talk about more friendly fungi. Hello everybody, my name is Martin. I'm a French student in master degree from Bordeaux University. My internship takes place in the Explory program, which funds innovative scientific research addressing raptor topics, such as the FunGene project for fungal genetic indicators. This project focuses on the conservation genetics of ectomycorrhizal fungi. Ectomycorrhizal fungi are the primary symbiotic partners of trees in temperate and boreal forests, and they have crucial part in ecosystem functioning, in terrestrial biogeochemical cycles, and they provide many other ecological services. We had yesterday very good presentations about this. The FunGene project hypothesizes that the genetic indicators provided by the global biodiversity framework are not suitable for these species. These indicators represent the genetic properties of populations and can be estimated from demographic data such as individual counts. However, there is always an however, the ectomycorrhizal fungi are predominantly underground species with complex life cycles involving both sexual and asexual reproduction, which results in the presence of haploid mycelium, which can merge to form eukaryotic tissues, and also in the presence of partial clonality within populations. These features make it difficult to distinguish individuals and population, and genetic analysis is required to define genets which are genetic individuals. So genet 1, genet 2, genet 3 are different genetically. Our project will test whether the global biodiversity framework indicators are suitable or if new indicators are required to link demography data such as the number of fruits bodies, and genetic parameter estimates. Until now, on fruit bodies, on the genotyping, the easily sampled above-ground fruit bodies. However, the presence of these above-ground fruit bodies does not necessarily reflect the entire ectomycorrhizal fungi population, as a single mycelial genet can produce several to no fruit bodies during the sampling periods. Therefore, to comprehensively assess the genetic diversity of these fungal populations, it is crucial to sample both the above ground and below ground compartments, above ground with fruit bodies and below ground with mycelium and mycorrhizas. Consequently, this project introduces an innovative approach, genotyping underground individuals from soil and root tip samples. Soils contain mycelium and root tips contain mycorrhizas. In our project, we extracted the DNA from fruit bodies of two common European Basidiomycete species, Amanita citrina and Swellius bovinus, which are widely present in the pine tree forest ecosystem of southern Atlantic coast of France. In total, approximately 120 fruit bodies of Amanita citrina and more than 200 of Swellius bovinus have been harvested in 4 sites. To ensure these fruit bodies belong to the target species, they have been identified by sequencing the ITS region, which is the internal transcript spacer region of the ribosomal DNA, which is highly conserved within species. The 2 species have been subjected to whole genome sequencing in order to detect the microsatellite loci and to select the most pertinent ones to develop SSR-seq markers. About the genetic markers, we have chosen to use the microsatellites, also called SSR for short sequence repeated, because of their high polymorphism, which allows to distinguish individuals. They also enable the distinction between heterozygous and homozygous loci, making them ideal for studying the eukaryotic tissues of the basidiomycete species. And additionally, their species-specific nature is crucial to amplifying the DNA of target species from environmental DNA in root tips and soil samples. We have decided to genotype the microsatellite by sequencing to prevent for size homoplasy and to liberate the polymorphisms within repeated and flanking region sequences. So these markers will allow us to genotype the food body, which is the above ground part of the population, and to genotype the underground individuals. We have collected soil cores at a depth of 30 centimeters and mycorrhizas and root tip samples containing mycorrhizas, as you can see in this beautiful picture, from the same location as fruit bodies. For each target species, 30 soil samples and 30 root tip samples have been harvested. We extracted the total DNA from these samples to obtain environmental DNA, from which we expect to amplify the DNA of the target species. So that will allow us to reveal the underground genetic diversity for this species. In terms of further analysis, ITS meter bucketing of soil and root tip samples are planned to verify if the possible failure of amplification of some samples is due to the absence of the target species in these samples. The lab work for the SSR sequencing is still in progress, so I'm so sorry, but the results are not yet available. Sorry for the deep frustration. Through this process, we want to determine whether the above ground genotypes can be redetected in below ground samples, so soil and root tips, whether the genetic diversity matrix computed from below and above ground samples are correlated, and finally, whether the observational data from food bodies abundance correlates with the genetic matrix. As another question, we are also investigating which underground compartments contain soil or root tips containing the highest genetic and specific diversity for ectomycorrhizal fungi species. So to provide a first an initial answer to this question, we use an already existing ITS metabucketing dataset of soil and root tips from another forest. So these results are from this dataset. The first graph shows the mean number of ectomycorrhizal fungi species by sample for soil and root tips, and the second one shows the mean number of reeds associated with the species by samples for soil and root tips. As you can observe, soil samples revealed a greatest number of species, while root tip samples had fewer species but more reeds associated with them. That suggests that roots may act as a filter, selecting for certain species, while soil contains a richer but more diluted species pool. In our study, we will test whether the high species diversity observed in soil samples is correlated at the intraspecific level, or whether the root tip samples reveal the higher intraspecific genetic diversity than soil samples in our two target species. To conclude, finally, Answering all these questions will be crucial to develop appropriate tools for ectomycorrhizal fungi conservation genetic assessments and to better understand the link between food body, mycorrhizas, and mycelium dynamics. I mean spatial and temporal dynamics. A major limitation of this study could be the potential lack of strong correlation between demographic parameters from food body and the genetic parameters, which could prevent us from developing appropriate genetic indicators for this species. Thank you very much for your attention.
Many thanks, Martin, for your contribution, and thank you very much to all speakers for your productive and interesting presentation. Now we have time to discussion, so I would invite to question from audience. And are there any question to our first speaker? Please, Sofia. Her presentation was about understory regeneration. There are any questions?
Hi, are you consider the light access to the forest floor? Because with this pine regeneration, because pine is very sun-demanding species, maybe there was more light access at that site.
Yeah, we measured, thanks for your question, we measure light access by hemispherical photographs, and so we measured canopy openness, and there wasn't any significant difference between ancient and post-agriculture forests regarding to light access. Is that your question about?
Yes, and I have a similar one because it's a highland. The topography is very diverse. So do you also consider the slope and the topography differences?
Yes. Regarding to elevation, the plots, all plots from post-agriculture and ancient forest were comparable in case of elevation. In case of slope, because much bigger slope steepness general in ancient forest than in post-agriculture forest, we performed the sampling for research plots in we divided in the area in for slope steepness. Sorry, how to? Yeah, to make it comparable, we perform not maybe general random sampling, but random sampling in research with comparable slope steepness. Yes. So we divided into 4 ranges of slope steepness to make it more comparable. So general post-agriculture forest slope steepness was comparable with ancient forest slope steepness in our research plots.
I know it's always challenging for that type of the research to find these similar sites.
Yeah. Our most important aim was to make these plots as comparable as possible.
Yes. Yeah. It was a very nice presentation, very good
Thank
research.
You.
Somebody else?
Thank you very much for your answer. And okay, please.
Thank you for your nice presentation. I have a question. You considered a different factor, but if I remember well, you didn't consider like some bioclimatic variables such as precipitation, temperature. And that's my question, why you didn't consider these factor?
This region is rather difficult to access and difficult to, this all my all research plots were far away from roads and in the middle of the forest, so it was difficult to put them measure. It was difficult to come back there later, so our aim was to access it once and don't come back there later to do all measurements, because to measure precipitation, for example, we have to leave there leave them the stuff to measure it. So so we decided to measure this component connected to climate by compound topographic index, which is about moisture, soil moisture, and heat load index, which is about exposure and it's about light, access to light and sunlight and exposure.
Okay, the point is that for the climate data, you don't need to put some equipment to measure it. You can, I think, easily to get the closest synoptic data source.
But they are too general to compare it between these points, these plots. Available climatic data are too general to compare this data between these all research plots.
And I have one more question. You mentioned about distance from the ancient forest. It's my question how the distance from the ancient forest could shaping the diversity on the post agricultural?
Because this is the source from ancient forest species can migrate to post-agriculture forests. So if the distance is closer, the regeneration is quicker, because these ancient forest species can migrate, because they are dispersed mostly by ants, for example, so they spread to short distances. So if the distance is long, it's difficult to spread for this ancient forest species.
And how the other variable could affect, like you consider like a slope, elevation, and so on. The correlation, I mean, could share effect of because the distance, when we are talking about the distance, we consider also the barrier. So did you consider also those elements?
Your question is if I consider the slope and elevation?
Yes. The barrier, how could affect also the distance from the...
this barrier. I think we also consider elevation and slope, of course, but you mean if we consider it as the slope as a barrier to...
Yes.
It is good question. I think it would be difficult to measure it as a barrier. What exactly is it?
Because it's like an interrupt, the barrier, like a topography. It's like a barrier, okay? So to make an interrupt to the like a gen flow.
But that I think that this. For example, steep slope is not a barrier to spread ancient. Yeah, that's true.
But I think also the river is like a two-way. Also could help to like flow the.
It's not one simple answer. What is the barrier? So it would be difficult to measure what is exactly this barrier and how to define it. So but it's good to think how to define it and how to measure the influence of this barrier.
And sorry, I have one more question. You point about the connectivity, and have you checked how the connectivity could shape the post-agriculture? You have an ancient forest, so you said through some years you get this post-agriculture. Have you checked how the connectivity could affect of this procedure?
But you mean connectivity with ancient forest, or it's? I think it's similar measuring the distance to the ancient forest. So,
like find some connectivity point. I mean.
I think it would be so similar measures because most of these post-agriculture forests were close to the border with ancient forest. I think there maybe I'm not sure right now, but I think that there were no such situation that the border from the ancient forest were far away, or they're mostly connected with ancient forests, with from one side at least. So I think this measures like connectivity and distance would be too similar to treat them like. I think it would be like, to treat them separately.
Yeah, I agree with it because in my opinion, they are completely different things. That's what my question. But we can discuss about it later.
Yeah,
after.
Of course.
Yeah,
but
it was very nice topic, and I was impressed. Thank you.
There were also like younger forests, younger post-agricultural forest. Yeah. And that younger post-agriculture forest were located farther from ancient forest. But that post-agriculture forest, which have developed just after the displacement, they were connected to ancient forests with at least one border. So they are mostly connected with ancient forests.
Okay. Thank you.
Many thanks for discussion club. Any further question to Sofia? No? Why? Thank you very much. And I would like to invite our second speaker. Please, Natalia, your question to Natalia. One second.
First of all, very good presentation. I want to ask you that maybe you know about some other places where this active protection was implemented successfully already.
Mostly in center and west Poland, we have also places where the thermophilus oak forests are actively protected, and we have a different type of management. And the studies shows that whether we use thinning cutting or animals to grazing the forest floor, we have the same effect. And the effect is, of course, positive for the alpha diversity and for these rare and protected plants. But there's one problem with grazing, because if it's too intensive, it develops the growing of grasses species.
More ruderal species, maybe.
Yeah, maybe more ruderal, and the grasses area is growing, And it's like physical taking place that other plants should take, but it's a marginal problem. But like I said, in Poland and in Czech Republic, in Romania, we have Thermophilus oak forests actively protected, and the results are very, very good. So we should keep doing it and apply in other places.
Which species from those forests are your favorite?
My favorite is Lilium martagon, and it's my favorite plant from Thermophilus woodlands, and my favorite plant of all wood species. And this plant is also considered as one of the most beautiful from Poland plant lists. So yeah, Lilium martagon.
So thank you very much.
Many thanks, Natalia, for your answer. And other question? Okay, please.
Thank you for your nice presentation. I have a question. You talk about the local hotspot. Have you think about it? How could we find those hotspot places?
By the local hotspots, I mean the tree fall gaps that happen naturally because of the loss from the stand. And these trees are growing in special conditions where they are able to develop a big crown. And when they fall from the stand, there is a huge gap. So this is like a nature gave us this local micro hotspot. So when we remove the wood from there, but because it's also an important thing in active conservation of this type of woodlands, that we cut or take the wood that came from the natural fall of trees and take it out from the borders of this place to prevent from the nutrification of the soil and the high topo. I forgot the word, but you know what I mean. Yeah. So to find and identify this local hotspot, we can use also a LIDAR system or other geo information systems. But in this case, actually the other step of my study is about the dynamic in these local hotspots, in these gaps. And we just find them by going into the woods. And we want to also make measures like relief and temperature and light index, and to compare these gaps and the close stand fields.
And those this local hotspot, it's no matter is like natural or done by.
No, it doesn't really matter. But I think it's the best when we just listen to the nature and we can try to mimic the process. But when process is already happening, like in this stand has Populus tremula trees. Around 120 years, that's the border for them, and they are going to fall right now, right in these years. It's happening, and it's still going to happen. So the nature gave us the process, and now we can use it. But also, in my opinion, we should cut some of the oak trees because they are like too near to each other. And they're growing up, not thick.
Yeah. Okay.
And to keep this, type of forest in the shape, we should also consider cutting oaks to give them the space to grow thick.
Okay. Thank you very much.
Thank you very much for your questions.
Thank you very much. Any question to Natalia? Thank you very much, Natalia. And I would like to invite our third speaker, Ioana. Please, your question to Ioana. Mm-hmm.
I don't need the microphone.
Mm-hmm. Please, Dr. Francesca Latterini.
Okay. So, very nice presentation. I admit my complete ignorance in the topic of fungi, so my question could look like a bit silly. But of all the species that you check, that you found in the Wrocław Park, which of these can be considered dangerous, for example, for the stability of these trees, for the mechanical stability? Because in the park, one of the most important factors is that these trees have to stand and not fall down on the end of someone. So which of these species could be dangerous about this?
I think the most dangerous one would be the one from sweet chestnut, because it's already described as like it causing a disease also to the leaves as well to the fruits and to the twigs. The rest of them is kind of some of the first reports of those three species. So it's right now it's hard to assess how they will impact those specific trees and as well the other species that are around, because they can spread to different one. But I think the one from sweet chestnut, as it's the most described, and by now we can say that it can be a problem, as I said, especially for this small part of this park, because this is like point when they put all of the sweet chestnuts in one place.
Thank you.
Which one of these species, or if there is any among these species, do you think could cause some very serious damage in many different areas? Do you think there is such a one?
Again, the one from Castanea sativa, but also I would like to check more on this, the last one, the Apiognonomia hystrix, since it was found before in Poland on the maple trees, and here is a potential for spread for other maple trees in the park. It's kind of not assessed how much of the trees are already infected with this fungi, and so it's kind of something that should be looked into for this one. All of them can impact different trees in different ways, so I would say all of them are important to monitor.
Okay, I have another one. So as we are having some big problems still with other introduced plant pathogens such as ash dieback and Dutch elm disease, and if there was such a disease to start, do you think there is any way to stop it early?
I think for plant diseases, the best way is to prevent them. So to monitor, like observe the trees and look for the symptoms before the disease spread widely. So the protection and prevention is the main way we can do right now with emerging pathogens. That's why we have all of the phytosanitary controls on the borders. That's why the plants have passports. So we try to protect ourselves from those, from spreading of those species.
Thank you very much for your question and answer. Any question to Jana? Okay, and I would like to, thank you very much, and I would like to invite our final speaker, Martin.
Everybody's hungry? Mm-hmm. Oh, yes.
Thank you. Please.
Yes.
Please, Professor Marcin Petras.
I have one question about why you select those two guys, because I understand that celluloid fungi are perfect for this such study because they form a lot of ectomycorrhizas and form a lot of extramatricial mycelium. But why Amanita citrina? Because I think that this species is not so abundant and frequent in the soil, and I'm working with ectomycorrhizas, I don't know, maybe 20 years, and I'm almost sure that I haven't seen ectomycorrhizas of Amanita citrina.
Interesting. It was because of the availability of the fruit bodies, which enable the development of genetic markers. And we succeed in amplify DNA of these species in a root tip sample. So that means mycorrhizas are present. But in fact, the first driver of our choice was the availability of fruit bodies, but other species are included in this study. But for the moment, we just want to test the methods. Good, thank you.
Please.
So I have many times been told that with these ITS meta-barcoding studies, you always get a lot of different fungi, and many of them may be dead in soil, right? And do you have any way device to tell apart which might be alive and which might be dead?
In fact, in the case of meta-barcoding, this could be a problem. However, here we work at the intraspecific level. So, if we found an allele from a dead individual, it may be a problem. However, this individual have existed, so that give an idea of a global genetic diversity. But in fact, from work at the community level, this is a problem, and we can use, for example, the abundance of reeds. If there is only very little quantity of reeds for a species, maybe it's not a good idea to take this species in account. Thank you.
Yes, I have another one as an avid mushroom picker. Now, I won't take any amanitas, but I do pick a lot of suilus. And when you do such a study, do you have any problems with foraging?
With what, sorry?
Foraging. You know, someone goes to forest and takes all your mushrooms.
Ah, yes. Initially, there were more places, more sites, sorry. But in fact, we have to throw some of them because no fruit bodies, no enough. And it was an idea to develop link with people who already know the spots, but they don't want to share it, of course. So it was complicated sometimes, in fact. Thank you.
I also have one question.
Yes.
What do you think? Relationship between soil and root trip genetic diversity would remain stable across broader environmental conditions? For example, different forest types or climatic conditions?
Good question. Previous study, I don't have a clear answer. But previous studies show that mycelium and fruit bodies, they do not have the same answer to the variation of climatical conditions. So, for your question, I don't really know what to say. Sorry.
What do you think? Your opinion?
I don't know.
Answer from audience. What do you think? Maybe Professor Marcin Petrans. Okay. Many thanks for all speakers, to all speakers, and I invite everyone to continue our discussion during lunch. Thank you very much.
Thank you.
Okay, welcome everyone at this afternoon session. My name is Daniel Chmura. I will be hosting this session. Before we start, I have two announcements from the reception desk. Actually, these are reminders. Tomorrow breakfast starts at 7, and you leave for the institute at 8:15. I will remind that also at the end of the session. Maybe someone will join us. Okay, our first talk in this session is talk by Agnieszka Hutniczak from University of Silesia in Katowice. Please. Sorry, just two organizational things for the speakers. 2 minutes before the end of time, I will raise my hand, and 1 minute before the end, I will just rise so that you know that to approach the harbor.
Thank you. Good afternoon, ladies and gentlemen. Thanks for coming along. It's great that so many of you were able to make it this morning. Let me introduce myself. My name is Agnieszka Hutniczak, and I am from Silesian University in Katowice. At the beginning of my speech, I would like to thank the organizers who allowed me to give the presentation. It gives me enormous pleasure indeed. The presentation is connected with the recent interest of our team and is entitled Micromorphology of Epicuticular Waxes and Cell Wall Chemical Composition of Aspen Leaves, Populus tremula, from Novel Ecosystem Habitats. So let me give a speech outlining the most important issues. Ladies and gentlemen, first, I will start by introducing the topic. As we all know, the rapid development of civilization, including urbanization, and the constant demand for natural resources have led to the transformation of the environment. These altered habitats, no longer resembling natural or semi-natural habitats, create, based on new non-analogous species composition, novel ecosystems. These habitats are inhabited by plant species that have the ability to adapt to this often difficult habitat conditions. Examples of novel ecosystems include coal heap, zinc heap, lead heap, quarries, as well as unused railway tracks. But, ladies and gentlemen, why should we study cell walls at all? Cell walls are integral, metabolically active parts of the cell, dynamically responding to stimuli from within the cell or the environment. Cell wall can be considered as a two-phase system in which cellulose microfibrils are surrounded by amorphous matrix, primarily composed of hemicellulose, pectins, proteins, phenolic compounds, and water. It is difficult to imagine that the waste surface area of higher plant bodies in contact with nature. It has been calculated that this is 2,100,000,000 square kilometers. So it is 8 times larger than area of all continents put together. This surface is made up of inconspicuous, rather thin, but crucial structure for plant, epidermis. Due to this, plants communicate together with the outside world, and it is possible to answer to various physical and biological factors. The characteristic feature of epidermis cell is the presence of cuticle. In turn, the cuticle is usually covered with epicuticular wax. So in our research, we examine both the leaf surface, analyzing epicuticular wax, And we also look deeper into the cellular level, focusing on cell wall immunohistochemistry. So the aim of our research is to analyze the epicuticular walks and compare the chemical composition of cell walls of plant leaves from novel ecosystem habitats and a control area. Cuticular walks is one of the two main components of cuticle. We distinguish epicuticular and intracuticular walks. The difference between them is connected with the localization within the cuticle. Wax is the main barrier preventing extrastomal water loss. It helps protect plant surface from pathogens and ultraviolet radiation. It influences plant-insect interactions, and wax is one of the mechanisms of adaptation to drought stress. Many attempts have been made to classify this diversity. The most commonly used classification is that proposed by Bartrott et al., based on the analysis of approximately 15,000 species from all major groups of seed plants. It includes approximately 20 types of wax formation grouped into three main groups: films, layers and crust, and crystalloids. The figure on the slide shows examples of wax crystalloid forms, for example, granules and platelets. And now, the second use technique, immunohistochemistry. It is the technique used to visualize the presence of a specific antigen in cells' tissues by using a specific primary antibody that binds to it. This technique allows us to examine the localization of pectin epitopes and arabinogalactan proteins in plant cell walls. Due to the fact that the chemical composition of plant cell walls in relation to environmental conditions is not sufficiently known, an analysis of these issues has been undertaken. I would like to draw your attention to the fact that pectins play an important role in modulating cell wall structure in response to different stresses and in control of plant growth and development. In turn, arabinogalactan proteins participate in many fundamental processes of cell growth and development and perform signaling functions in cell walls. The most important functions are highlighted on this slide. The material was collected in 2022 growing season randomly from spontaneously formed patches of vegetation dominated by studied species, overgrowing the post-industrial novel ecosystem and control site. Studied areas are marked on the map, and the photos illustrate the studied types of habitats. A prerequisite of the study was the assumption of different nature of biological processes on types of sites taken into account. Before beginning to analysis, it is very important to collect plant material. Plant material consists of leaves from Populus tremula, which is a common deciduous tree. It is a pioneer species with high ecological plasticity. The next slide shows the stages of epicuticular wax analysis. So, as you can see, after air drying the leaves, we cut plant material under a binocular and place leaf fragments on aluminum stubs using double-sided carbon type. The next step is coating the samples with gold using high vacuum sputter coater. We analyze the prepared samples using Hitachi high-resolution scanning electron microscope equipped with the EDS X-ray microanalysis system. And now I would like to show the most important stages of immunohistochemical procedure. covering fixation, deaeration, dehydration, and embedding of plant material in accordance with generally accepted immunohistochemical procedure, working with a microtome, incubation of slides with primary and secondary antibodies, and the last stage is visualization of immunohistological staining at the light microscopy level. So we can say that it is a long procedure. Now, I would like to show the selected results. So, as you can see on pictures on your left, you can see leaves from hips. And pictures on your right are collected with leaves from control site. So now I would like to show comparison of the structure of the epicuticular waxes of the upper surface of Populus tremula leaves from the novel ecosystem habitat and a control site. Analyzing the wax, we can say that it exhibits different microstructure. It differs in size, shape, and the density. In the case of hips, we can say that epicuticular wax dominated in the form of crusts. In turn, in the case of control site, epicuticular wax dominated in the form of crystalloids. And now, I would like to present the comparison of the structure of epicuticular waxes of the lower surface of Populus tremula leaves. So, in the case of hips, we can see more crusted layers, which are larger, and crystalloids are larger and more numerous. In turn, in the case of control sites, we can say that crusts are less widely distributed and are smaller, and crystalloids are smaller and less numerous. I would like to emphasize that this research is still ongoing. Now, the differences in the localization of arabinogalactan proteins in Populus tremula leaf. So, you also can see the picture in your left are connected with the hips, and the picture on your right are the control site. So, in the case of hips, we can say that the signal was present in upper and lower epidermis and in vascular bundle. And in the case of control site, the signal was wasn't. We now observe the signal, and in the case of differences in the localization of low estrified pectins in Populus tremula leaf, we can say that the signal was visible in upper and lower epidermis and in spongy parenchyma. In turn, in the control site, we can say that the signal in epiderma was less intense and the signal was present in spongy parenchyma. So, ladies and gentlemen, in conclusion, I would like to draw your attention to the fact that the research on plant responses to stress factors has been conducted primarily in laboratory conditions. However, the knowledge of the adaptability of species to extreme conditions is crucial. Various research methods covering organismal, subcellular, and even molecular level should be carried out. So, to achieve the understanding of functioning of the entire ecosystem, interdisciplinary research should be carried out. Undoubtedly, plant studies at the cellular tissue level can be an additional source of knowledge and an excellent complement to analysis conducted at the level of the whole organism and the ecosystem. I would like to thank everyone who helped me during my work. I would like to thank especially the leader of our team, Professor Gabriela Woźniak. In the end, I would like to show the list of selected publications relating to the discussed subject. Thank you for your attention, and I kindly ask you for cooperation. Thank you.
Thank you very much for your talk. Our next speaker is Beata Ciak-Wojdyła from the University of Rzeszów. She's going to tell us about forest understory plant responses to increased light and temperature.
Hello everyone, my name is Beata Ciak-Wojdyła. I come from the University of Rzeszów, where during my PhD, and I'm researching the impact of increased temperature and light availability on a forest herbaceous plant, for example, Galeopdolon luteum. And today I will present how light and temperature interact to shape the physiological response of forest understory plants. The first question is why is this important, exactly this interact? Forest understory plants naturally grow under low light condition, but even small changes in canopy structure can increase light availability. And at the same time, rising temperature factors modify the microenvironment at the forest floor. Understanding how plants respond to the combination of this factor is critical because their interaction may strongly influence plant functioning and ultimately forest ecosystem dynamics. In this study, we wanted to understand how increased light availability and elevated temperature affect the functioning of forest understory plants. We focused on both structural traits and physiological process, including photosynthesis. Importantly, we looked not only at the individual effects of these factors, but also how they interact. The main objective of this study was to assess how light and temperature influence plant functioning of the forest understory. And the hypothesis, the first is light and temperature significantly influence plant functional traits and photosynthetic efficiency. And the second, their interaction modifies plants' response. And let me start with why we choose this species for our research. On first is common in deciduous forest herbaceous plant in deciduous forest almost all over Europe and small part of Asia. It occupies a significant part of the forest herbaceous plant layer, which plays an important role in the functioning of the forest ecosystem, for example, the proper circulation of water and elements. To evaluate plant response, we measured several groups of parameters. We analyzed leaf functional traits such as specific leaf area, leaf dry matter content, relative water content, and we also assessed photosynthetic performance as well as the content of photosynthetic pigments, chlorophyll A, B, and their ratio. We conduct our research in a specially breeding room, which we see in the photos, with constant temperature, lighting, and controlled humidity. In the first photo, I show initial breeding, and in the middle and the last photo is proper breeding Galeopdolon. On the first steps, vegetative shoots of the Galeopdolon luteum were collected in designated part of the oak hornbeam forest near Rzeszów. These shoots were then divided into small parts and placed in sand as in first photo. And after 3 weeks, they were transplanted into pots as in second photo. And about 300 individual plants growth 2 months in a breeding room under climatic conditions reflecting those in forests of Central Europe growing season, 21 during the day and 13 during at night, with a photo period of 60 hours of daylight and 8 hours of darkness, and these were the controlled condition. This graph shows the design of the experiment. First, plants were grown under controlled conditions for 2 months with low light and low temperature. Then they were exposed separately to higher light or elevated temperature at 25 and 29. Importantly, plants under high light had been growing in pots under condition from the beginning. In the final steps, both factors were combined to assess their interaction. Samples were collected at several times points during the first day and again after 1 week. And we go to the results. Our results show that increased light availability led to distinct change in leaf structure, including higher leaf dry matter content, suggesting the development of more robust leaves. In here. Performance index also increased, but its value under highlight was determined by temperature. At the highest temperature of 29, decrease was observed, suggesting limitation in the functioning of the photosynthetic apparatus. Increased light availability led to a decrease in specific leaf area and relative water content, and these changes were primarily determined by light intensity. This indicates a shift toward a more conservative leaf functioning strategy, including the development of thicker and more compact tissue and reduced water loss condition in increased radiation. The interaction of high light intensity and elevated temperature significantly influenced the content of chlorophyll A, B, and B, as well as the ratio. With increasing temperature and increasing with increasing temperature and increasing chlorophyll A content, which increasing temperature and increase in chlorophyll A content was observed, while chlorophyll B leaf increased at 25 and decreased significantly at 29. And these changes led to an increase in the chlorophyll A to B ratio, indication a remodeling of the antenne complex and reduce in light absorption capacity. We observe a clear shift in how light and temperature interact. Under moderate conditions, they work together and support photosynthesis. But as temperature increases, this interaction become less beneficial and may start to limit plant functioning. This pattern is also reflecting the effect size analysis, which we see in this free graph. This pattern is also reflecting in the effect size analysis. Light is the main factor driving changes in specific leaf area and photosynthetic efficiency, as shown by the highest effect size value. In contrast, chlorophyll B is more strongly influenced by their interaction between light and temperature. This suggested that while structural traits are mainly controlled by light, the photosynthetic apparatus is more sensitive to the combined effects of environmental factors. This is important because forest understory plants are highly sensitive to environmental changes. In natural conditions, increased light often occurs together with higher temperature, for example, after canopy disturbance. These combined effects may influence plant survival, species composition, and overall forest ecosystem functioning. These results suggested that light acts a key driver of plant response, inducing structural and physiological adjustment. However, temperature modifies this response, especially under high light condition. And conclusion, light availability is the main driver of leaf functional changes. Temperature modifies plants' response to increased light. Their interaction strongly affects photosynthetic performance and chlorophyll content. And the last, these results highlight the importance of combined environmental factors in shaping plant response under changed climate condition. And thank you for your attention.
Very much. We have even time for one or two questions. Please stay here because your talk was so effective in time, so we still have time before we should move to the next talk. So are there any questions to this presentation? Well, actually, I have one question. You showed us the light levels you used in your experiment. If I'm correct, these were 11.8 and 64.4 micromoles per square meter per second. Is that right? I mean, this 64, it's still relating to open light. It's still low, so why do you consider that as a highlight treatment?
This highlight for forest understory plants.
All right, fair enough. Okay, are there any other questions? We still have, what time is it 40 already? Okay, so maybe we'll, thank you so much. We'll move to the next talk. Our next speaker is Mariola Rabska from Institute of Dendrology. We move to Diawesi again from yesterday talks.
Thank you. My name is Mariola Rabska and I'm from the Institute of Dendrology, which I think you already know, this institution. And today I will talk about some part of results we obtained in our research concerning intersexual differences in common mistletoe, Viscum album. And first thing, what Viscum album is. It's a hemiparasitic species. So it can photosynthesize, but it's partly dependent on the host trees or shrubs it grew on. And in Poland we have 3 subspecies of this hemiparasite, and they are different in the way they colonize different species of hosts. And the subspecies we analyzed, Viscum album subspecies album, colonizing deciduous trees and shrubs. And it's interesting because this chemiparasite is evergreen, so it can photosynthesize even when the host tree is dormant. And the most important thing about this species in the light of our research is that it's dioecious, so we have separate male and female plants. And previous studies confirmed that a lot of population of this species show female-based sex ratio. And when we have male and female plants, we can expect some obvious differences between them concerning flowers. But we have, in a lot of dioecious species, we can observe also a secondary sexual dimorphism, which is not directly connected with reproduction, but it's indirectly connected with different roles played by plants. And more than 100 years ago, it was observed that male plants of Viscum album are more yellow during spring than female plants. But it was only an observation and it wasn't confirmed chemically. Our research were conducted in the Arboretum of the Institute of Dendrology, and as a host we chose malus trees, and those apple trees were quite old because the oldest were almost 100 years old, which was important because Viscum album preferably colonize old trees. We measured all those trees and they were very variable in size. And the first thing we wanted to check was if those observations about color of male and female individuals can be confirmed on the level of chlorophyll concentration and carotenoids. And the other thing was that we were wondering if male and female differ in the position of the crown they colonize of the host tree, and because it could be connected with more light availability, and then they can be different in color. And we also wanted to check if those more green leaves of females can be connected with higher photosynthesis, higher sugar production, and in consequence females could be larger. The first step of our research was to count all the mistletoe clams growing on the population of apple trees, and then on about 100 apple trees, we measured all the clumps in details. So we specified their age, sex, and we measured clump wideness, how high the clump was, and the length of the longest shoot. And we also measured the distance between the clump and the ground. Besides of those field measurements, we also measured photosynthetic pigments in laboratory, and it was conducted in 2 stages, because on the first stage it was some kind of preliminary studies. We measured photosynthetic pigments in March. It is a time when those differences between male and females are the most visible. And when we found some differences in photosynthetic pigments, we decided to conduct a second stage of our research, and we wanted to check the differences between sexes in a longer period. So we measured them from August to June, every 2 months for 1 year. And in those analyses, we identified the concentration of basic photosynthetic pigments, and we counted basic ratios concerning this. Our population contains almost 900 individuals. And we observed that in our population, like in populations which were counted by other researchers, we also observed a female-based sex ratio in our population. And what is important in the light of our research is that the age structure of both sexes was similar. And the clump size was greater in females in all 3 parameters we measured. They were wider, higher, and they have longest shoot. And we didn't find differences between sexes in the distance from the ground of the clump. And what about photosynthetic pigments is that we observed that both chlorophyll A and chlorophyll B was higher in females. And this trend was observed from December till April in the chlorophyll A and even June in the chlorophyll B. A similar situation was for total chlorophyll, which is not surprising. What was interesting for us was that chlorophyll A to B ratio was higher in males, both in our preliminary studies in March, but also in February in our long-term studies. So it was more or less the same time of the year, and this ratio was different between sexes only during this short period, during the whole year. And what was surprising for us that carotenoids were not higher in males, because we expected it, seeing that they are more yellow. But we didn't find this trend in carotenoids. But we found that most of the measuring terms we analyzed, in most of those terms, females had a higher chlorophyll to carotenoids ratio. And going to the conclusion, we proved that those observations about differences in leaf color between male and females are visible on the pigments level also. But we think that the key parameter which shows us those differences is chlorophyll to carotenoids ratio rather than simple carotenoids concentration. And what was interesting was that the ratio of chlorophyll A to B was higher in males And it was during only a short time of the year. And at this time of the year, the host of those trees is of Viscum album is dormant, or just break the dormancy, and at the same time Viscum album is flowering. So we think that males have higher, this ratio in males is higher because they have greater effort connected with reproduction during this period. And it suggests that they are more stressed and they have different resource allocation strategies. We didn't find that the connection between pigments and the position of the clamp. But what was interesting is that we found that we just not found the female bees sex ratio, which is common in literature, but we also found that females are larger. And what is important is that it wasn't connected with age, because we didn't find differences between sexes in age structure. So it wasn't that females were bigger because they were older. It was caused by other things, maybe those greater concentration of photosynthetic pigments. But it's still open question why those trends were observed by us, and some of those results we obtained were already published, but we still have open questions concerning this interesting species. Thank you for your attention.
Thank you very much for the excellent time. Sorry, it doesn't work. For the excellent time, keeping time. We have time for one question, maybe, before we move to the next talk. No questions. That's yeah. Please.
This dimorphism, this difference between this male and female, is very interesting that you notice this. And it is also possible to notice the same type of the differences between other species. In other species, like, I don't know if I ask this.
You mean dioecious species?
Because
or
you
between
observe
species?
these differences in the morphology between male and female. We can also find the same differences between male and female in other species, plant species.
Yes, in dioecious species we have a lot of secondary sexual dimorphism, but in general we observe that females are more vulnerable for stresses, and we could expect that they would be smaller and more fragile. But in this species it's opposite, and we found that females are bigger and they
this is very
show.
unique.
Sorry.
How unique is this difference that female are bigger?
Once again?
How unique is this that females are bigger?
For this species, or
for the plants?
It's quite unique. We think that it's connected with hemiparasitism of this species.
Thank you.
So the females are probably more effective invaders. Thank you. All right, thanks a lot. Our next speaker is Yikazu from, I probably shouldn't try to pronounce this name, but I'll try anyway. Shishungwanba, sorry. Tropical Botanical Garden. Yeah, you can pronounce it properly. Sorry for that. Yeah, please welcome.
Okay, thank you. And hello everyone. My name is Ecology. I am a PhD student from Xishuangbanna Tropical Botanical Garden, Chinese Academy of Sciences. And I'm very glad to share my research here. And my research topic is autogenetic shifts in ecological strategy in a giant tropical tree species. No, it doesn't work. Oh, I can use this. Oh, thank you. My presentation will be in 4 parts. First, I will introduce my research background. During last decades, the frequency and intensity of droughts have increased along with rising temperature, decreasing relative humidity, and rising vapor pressure deficit. And drought-induced tree mortality and forest decline occurs more often. And this caused negative consequences on forest community structure and ecosystem functions in southwest China. And drought-induced tree mortality is a highly relevant topic in global change research. And numerous studies had showed that tall trees may have greater risk during droughts. And for tropical forests, they are generally tall and highly susceptible to drought. And they exhibit more severe hydraulic damage under droughts than shorter forests. And currently, there are 3 main explanations for why are tall trees at greater risk during drought. Firstly, based on the hydraulic framework, tall trees have higher metabolic requirements, making them more susceptible to carbon starvation when drought limits carbon uptake. Secondly, the internal hydraulic structure of tall trees caused a greater risk by greater gravity and asylum tension in water transport pathways. And they also have narrower asylum transport safety margins. Thirdly, the vapor pressure deficit amplifies the association between tree mortality and tree height. And tall trees have huge canopies. Those canopies was exposed to more drier microclimates and stronger solar radiations. Those reasons made it more increases its vulnerability. And functional trees are useful tools to investigate tree physiological adaptation to drought. For example, photosynthetic capacity represents the maximum functional potential of leaf photosynthetic systems. Maximum photosynthetic rate responds to droughts rapidly. And leaf tuberose point and asylum water potential at 50% loss of hydraulic conductivity are proxies for drought resistance of leaves and branches. And to some extent, the relationship between these 2 hydraulic traits can define the hydraulic strategy of this plant. And leaf branch allometry and Huber value indicate carbon investment in different organs. Within a species, functional trait values and trait relationships can shift with autogeny and environment. And for tall tree species, individuals at different developmental stages adopt different ecology strategies to cope with environmental filters and natural selection pressures. And besides only investigating the trait values of seedlings or mature trees, may mask critical shifts in plant development. So research based on more developmental stages are needed. And Paraschorenhinensis is a tropical giant tree species, and its height can reach 70 to 80 meters tall. And it is protected in China and also listed as an endangered tree species worldwide. Its habitat stretches in the northern edge of Asian rainforest. And Paraschorenhinensis plays important roles in tropical rainforest by maintaining biodiversity, fixing carbon dioxide, regulating climate, and providing timber. And so considering its important roles in tropical rainforest and given its potentially drought vulnerability, so we conducted this research. Here are my two scientific questions. Question 1, how does the functional traits of Paraschorenhinensis change with developmental stages? Question 2, how do eco-ecological strategies of Paraschorenhinensis shift during autogenetic tree growth? And here are my hypotheses. First is trait values related to carbon uptake initially increased with autogenetic development and then decreased, while values of traits related to hydraulic and mechanics consistently increase. And individuals of Paraschorenhinensis in the understory canopy and above canopy layers exhibit different ecological strategies. And next, I will introduce my materials and methods. This research was conducted in a Paraschorenhinensis-dominated lowland tropical rainforest in southwest China. And there is a canopy tower crane, and there has 80 meters operational height and 60 meters job length. And the climate there is distinctly characterized by a rainy season and a dry season. And we grouped the individuals of Paraschorenhinensis into 6 groups based on its tree height. So in this research, there are 6 autogenetic stages, including seedling, sapling, juvenile, young adult, mature, and old emergent tree. And we selected 6 individuals for each developmental stage as replicates. And to quantify the variations of microenvironment factors change across the first vertical environment, we measured 4 environmental factors at 6 height gradients. And different sensors were mounted on 3 Paraschorenhinensis trees in the forest. And we selected 25 functional traits and divided into 3 functional groups, including carbon uptake, hydraulics, and mechanics. And all the measurements were conducted in the same growing season. Next is the results and discussion part. This figure shows the variations in microenvironmental factors. As tree height increased, tree canopy was exposed to microenvironments with higher photosynthetically active radiation and vapor pressure deficit. And this is the comparison of trees related to carbon uptake among 6 stages. Maximum photosynthetic rate, maximum stomata conductance, photosynthetic nitrogen, and phosphorus utilization efficiency are increased significantly and reaching a maximum at the 5 to 15 meter stage and then decreased. And chlorophyll content decreased significantly. And this is the comparison of hydraulic traits among 6 stages. Branch water transport efficiency, leaf drought tolerance, leaf capacitance, and leaf water use efficiency increased significantly, but branch drought tolerance decreased slightly. And this is the comparison of mechanical properties among 6 stages. Mechanical strength of branches and leaves increased significantly. And this figure shows the correlation among traits and environmental factors. First, look at the lower left corner, there is a grid. The significant correlations were found between trait and between branch and leaf traits. This indicated coordination between different organs and different functions. And next, let's look at the right side. Those edges indicates the correlations between 25 functional traits and 3 factors. That represents that developmental stages and environmental factors jointly influence the variations of functional traits. And this is the principal component analysis results among functional traits at individual levels. And from left to right, the blue circle and the yellow circle represents the first younger stages. And the red, and the light blue circle represent the middle 3 developmental stages. And the dark blue circle represents the oldest developmental stage. So there are distinctly 3 different ecological strategies. Strategy 1 is low light adaptation and hydraulic safety. Strategy 2 is faster growing based on high photosynthetic rate and high water transport efficiency and high water use efficiency. And strategy 3 is robust structure. Next is the discussion part. First, by increasing leaf chlorophyll content, individuals in seedling and sapling stages were able to increase their ability to capture light under limited light conditions. Although photosynthetically active radiation increased across the 6 developmental stages, but the maximum photosynthetic rates didn't continue to increase indefinitely. And because for taller stages, the light was no longer the limitations. Instead, water transport become the new constraint. And that means taller trees have a long distance to transport the water through root to their canopy leaves to make their more difficult to photosynthesis. And at the same time, in upper canopy, the vapor pressure deficit there is higher. That means more drier. And taller trees presented a range of structure and functional adjustments to cope with drier microclimates, including higher efficiency and lower safety strategy in branches, and higher drought tolerance and water use efficiency in leaves. And in Parent-Shorenhinensis, it exhibits a classic efficiency and safety trade-off in their branches. And by increasing mechanical strength, the branches and leaves become more robust. That helps support the extraordinary tree height. Okay, next is the summary. Following the developmental stages, most functional trees changed as the trends described in the hypothesis 1, except P50 and chlorophyll content. For hypothesis 2, functional trait values vary with tree height, enabling individuals of different developmental stages to exhibit distinct ecological strategies. And the novelty of this study it's because it covers 6 stages of Parent-Shorenhinensis and systematically revealing its autogenetic shifts in ecological strategy. And in future research, I plan to explore the adaptive strategies by analyzing the variations in non-structured carbohydrates in leaves and branches to detect the effects of seasonal dynamics on these tree species. And this is the acknowledgement part. Thank you to Professor Yajun Chen and Professor Henrik Hartmann. And that's all for my presentation. Thank you.
Questions to the discussion at the end of the session. Our last speaker this session is Kaisa Laidi from the University of Tizi Ouzou in Algeria. We talk about influence of canopy structure and light on the regeneration of Numidian fir.
Hello everyone. My name is Lady Kaisa. I'm from Algeria. I'm a PhD student, and today I will present you part of my thesis work entitled The Influence of Canopy Structure and Light. Sorry. In regeneration of Numidian fir in the Babur forest. First, I will present the species. The Numidian fir is in conifer endemic to Algeria. It is strictly confined to the humid north slopes of the Babur and Tababort massifs due to its restricted, limited area of 500 hectares. Due to its restricted distribution, ongoing decline, climate change, and human disturbances, the species is classified as critical endangered in the ICN Red List. It typically grows in light altitude in mixed stands with other species like Cedrus atlantica, Quercus canariensis, Populus tremola, and Taxus baccata, and other species. Forming a complex canopy structure that strongly influences light availability at the forest floor. In this context, understanding its regeneration dynamics is essential for ensuring its long-time conservation. We hypothesize that light plays an important role in the regeneration of Abies numidica. We expected that seedling establishment is highest under moderate canopy openness when there is a balance between too much shade and excessive light. We also suggest that different canopy structure create a different light condition. Therefore, this study aims to identify the light conditions that are most favorable for the regeneration. In this map, you can see the localization of the study area in the northern Algeria, within the Park National of Babur and Tababort. We selected different locations across the forest to capture variability in forest structure. At each site, we established a plot of 10 meters by 10 meters. Within this plot, we collected hemispherical photography to analyze canopy structure and light conditions. In total, 13 images were taken, distributed across the different sites. To do this, we used the hemispherical photography with a smartphone to measure canopy openness and light conditions. The images were processing in air, and we collected the diffuse non-respective represent light availability under the canopy. We also counted the number or the density of seedlings around each plot. Then we used statistical models to analyze the relationship between light and regeneration. Our result, this is figure show the different stage we're processing the image in under air. Our result showed that regeneration follows a non-linear response to light. Seedlings density was highest under intermediate and high intermediate light conditions, with an optimum around 15 to 20%. When the canopy is too closed, regeneration is limited. In the other hand, when the canopy is too open, the regeneration decreases. We also found different between canopy tips. Regeneration was highest under mixed canopy, moderate and fair dominated stands, and lowest under the cedar dominated canopy. These results highlight the importance of canopy heterogeneity in supporting regeneration. Maintaining intermediate canopy openings can improve the natural regeneration of this species. In conclusion, this study shows that simple and low-cost methods like smartphone hemispherical photography can provide valuable information for forest management and conservation. Thank you for your attention.
I have working microphone. Good. Okay, thank you again for the excellent time regime. And so we can move to the discussion at this session. Maybe please stay. Please stay where you are. We'll begin maybe with questions to your talk. Please. No immediate questions. So what your results tell us maybe about the ecology of the species? I mean, is it consistent with what you would expect from this species? I mean, the regeneration in small canopy gaps. Yeah, I mean, as abies species, it's probably late successional forest species. So it's your results just confirmed that it's this. Yeah. Okay. Good. Any other questions? Please don't be shy. Okay, thank you so much. Thanks again. So maybe we'll start with the first talk in this session. Are there any questions to Agnieszka Hutniczak on her talk about waxes in poplar? I'm looking at. Yep.
So I grew up in a somewhat similar area to what you described, with lots of former mining sites and heap mountains and lots of turned over land. And I see a slightly different species there. For us it's Betula pendula, but I understand that populus is not that widespread in Estonia. And I started wondering about the biochemistry part of the tree leaves, because I haven't heard about anyone in Estonia investigating it. And the ground there, the heap and that stuff contained quite a lot of nasty stuff, such as heavy metals and so on. And do you know if the trees tend to take up a lot of the heavy metals and other potentially poisonous stuff, or how is it?
At the beginning you talked about Betula pendula. And Betula pendula, we also carried out this species. It was the first step of our research. because we wanted to concentrate first of all at trees, and after that we would like to check also another species. But come back to your question. Yes, plants can accumulate heavy metals, and at the first step of our research, we checked that in our habitats we have got different metals, heavy metals, and because we want to compare species from different habitats, and different habitats is for us habitats connected with different plots connected with different habitats. So, for example, heaps can be another habitats than quarries. And for example, also unused railway tracks, yes? So we can check the presence of heavy metals, pH, and also we conducted the samples of soil. Mm-hmm. So different aspects of analysis.
Okay, thank you. Any other questions, please, Aga?
Hello, thank you for a very interesting presentation. I have a question because we saw a huge difference with signals from albinogalactans. Maybe you missed something, but did you analyze the thickness of the cell walls itself?
Yes, we also conducted research connected with these parameters, but now we are during the analysis because we want to correlate this aspect and also another functional traits of species. Thank you.
Any other questions? Proszę. Go ahead.
I would like to ask you about criteria for selecting trees first and leaves second. Because, for example, age of leaves, I mean, position on shoot may be important for chemical composition of the cell wall, I think, and also on level of destruction of epicuticular waxes. Thank you.
Thank you.
Yes, at the beginning of our research, we wanted to think about it because it is important. We wanted to concentrate on the same leaves from, for example, from the place, yes, from trees, in the middle of branch, and in the leaves are in a good conditions. And we tried leaves randomly, but always we collected 20 leaves from plots.
Okay, thank you. And I have one last question to you. Can you conclude that those differences you showed in the wax layers between control site and your affected sites, could they be indicative of the tree response to the conditions, or could they be treated as maybe a proxy for some acclimatory response?
Yes, we wanted to ask for this question, but at this stage I think it is too early, because now we are checked only few leaves. So now we will prepare another samples and also check it.
Okay, thank you so much for your answers. And now we'll move to the second talk in this session by Beata Ciak-Wojdyła. Any questions? I have one. Could you maybe please step up here, because we are streaming this, so you could be visible on the...
Okay.
in the internet. Okay, my question regards the photosynthetic index. You showed this trait. I don't. Well, actually, what was that? Because there was no explanation. I mean, clear explanation. What you mean by the photosynthetic performance index or efficiency index?
Efficient.
Okay.
Efficient. Performance and efficient. We used to pocket P and measure performance index, and we show
So is it the maximum rate of photosynthesis, or what is it actually? Because I I didn't understand that.
It's too hard.
Okay, maybe we'll talk about that in the
okay.
after that. Okay. Any other questions to Beata? No. Okay. So questions to Mariola Rabska on her talk about the mistletoe. We had already some questions, but well, it's very interesting that those female plants are bigger, and maybe they are more effective in, I mean, invading the host plants. W ogóle są bardziej liczne w populacji.
This trend is observed in a lot of population in Viscum album, and it's very interesting why it's not 50% to 50% ratio, and those females individuals have also some physiological advantages which I didn't show, and it's the clue of our research to find out what happens that it's not a balance between sexes in this species.
Any other questions to this talk? Yeah. If not, we'll move questions to the coffee break. And now the questions to talk by Yikazu about the tropical trees.
Sorry, congratulations for your presentation. My question is, you did the data collection in one season, and you said there is 2 seasons. Do you plan to do in the other season and compare the difference, or?
It's a good question. Yes. This is because my topic, I just want to compare the ecological strategies of 6 developmental stages individuals. So I choose the same season. And in my future research plan, I do measure some like photosynthesis and non-structured carbohydrates and and water potential and some other functional traits in both in rainy season and dry season. And this experiment cost several years, from 2022 to 2025. Yes, I have 5 years. Okay, thank you for your question.
Any other questions, Marzena? You wanted to ask some question?
Maybe I have a question. But most probably it would be really strange question, because I assume carbon and water transport is really important. So I have a question. Did anyone checked the speed of the transport of sap and water in trees with different height and age? This is the same speed, or maybe it's quicker in those taller trees? You know, I'm just curious. Okay, the speed of the transport, how quickly water and photosynthetic products are transported in trees with varied in age and height. Did anyone analyze it?
It's a little bit hard to answer actually, because I yes, I only measure a maximum photosynthesis rate and water transport efficiency. They happen in different organs, because photosynthesis happen in leaves, and most water transport efficiency happen in branches. Yes, but they are a significant correlations between maximum photosynthesis rate and water transport efficiency. I can only say this, but others know. Sorry. And
yeah, I was going to ask about the trade-off between hydraulic conductivity and safety, and I think you showed that there is a trade-off between this. Yeah.
You want me to explain more to that?
Yes, we still have some time.
Okay. Yes. In my from my research data, I can see that there is a negative correlation between water transport efficiency and water transport safety. They are different. They are measured by different methods, and there is a trade-off. Because in a classic research was published in 2008 in a paper, they said in most of the vascular plants, existed this traditional trade-off, because it's difficult to find a plant have the high water transport efficiency and the high safety in the wild. But you can find some plants both have lower water transport efficiency and lower safety. It's more common. And most of the vascular plants, they will have this trade-off. Yes.
All right, thank you. Thank you very much.
Okay, thank you for your presentation. But as a mycologist, I must ask, what is the mycorrhizal status of this tree? And have you thought about checking the differences of microbiome at different stages of the development?
Yes, mycorrhizal. Because our research group more concentrate on plant ecophysiology, and I know there is a related paper have do the comparison of the mycorrhizal among different developmental stages of these tree species. I can send this paper to you. Okay. Thank you for your question.
Any questions? If not, thank you very much. I congratulate all our speakers on the excellent research they've showed us today. And please give the round of applause to all the speakers once again. I would like to remind you that tomorrow breakfast starts at 7 a.m. and you leave for the Institute at 8:15 a.m. And right now there should be coffee break, but unfortunately only attendants in room B have coffee break according to the agenda. Sorry, that was a joke. So we have coffee break until 4:15.
interesting one with some of the exotic species, as I believe, at least for me. I think we are supposed to start right now, so I'll invite the first speaker, who's Fernanda Neves Lima, yeah, who's going to tell us about the genetic resources of rosewood. So please, you may start.
Hello, everyone. I'm Fernanda Neves Lima, and I am a PhD student at the University of Bordeaux. And today I will talk about genetic resources for a sustainable rosewood essential oil industry in South America, insights from genetic, genomic, and chemical approaches. My direction is Dr. Miriam Wertz, with the co-direction of Nicolas Tanslinski and Daniela Flores from INRAE et CIRAD in France. So this is rosewood, this is Aníbal rosodora, popularly known as rosewood, in Portuguese pau rosa, in French bois de rose, and it's a Heliophilus evergreen tree distributed through the Amazon biome. So Amazon biome is in the north of South America, and you can find rosewood in all these points that are marked here. Rosewood is a hermaphroditic tree, and she has a very singular flowering period, because during the day she separates, opening the male apparatus at some point and the female at the other to avoid the self-pollination. No, I think it's too much. Rosewood essential oil is used in top products of perfume industry. I don't know if you know Chanel No. 5. It's a very famous perfume used by Marilyn Monroe, and it made with rosewood essential oil. And rosewood essential oil is very rich in a substance called linalool. It's also found in lavender products. So linalool is a unique substance that have different aromas based in the isomery position. So if you have the isomery in the right side, you have one aroma and one type of application, and if you have in the other side, the left side, you have another aroma and another application. So to extract this oil, you need to cut the tree and then break all the wood, and you process it by hydrodistillation, and you obtain the oil. And that's the reason because she was very exploited in the early 20th century, that now she's classified as in danger of extinction since 2014, and the trade is regulated by states and national governments. So you can see here that the exploitation was really high, but after they start to notice that there are not so many natural populations anymore and start to control this exploration, the exploration went down and the price went high. So the last data was from 2019, and the price of the essential oil kilo was $350. That's why the Territory Collective of French Guiana requested the start of a genetic resource bank. And he was established in Risquetu, which is in the center of Guyana. And it was made by the National Forest Office in France in 2002. And you have seeds from 6 provinces in French Guiana, and you have 450 sections. And this was a way to represent all the genetic diversity of rosewood in French Guiana, and also this plantation is the plantation that donate seedlings and seeds for all the other commercial plantations of rosewood in French Guiana. So that way you can also trace this the genetic source of the seeds you're using, because you cannot exploit not natural populations. You only can sell essential oil from commercial plantations. And rosewood is also a very complicated species because there is problems in the taxonomy. You have a possible species complex with other Aniba from the Larosia family. You have a not very explained ecology in the terms of distribution of the species and also the regeneration, because it's a variation of soils that you can regenerate. And it's like here you can see a lot of seedlings and they were growing into the water. And at the same time you go to the east and you find rosewood in a sand soil. So it's very complicated to understand this. In the chemistry part, you have different chemotypes based in different oils from different places. You have the influence from the environment that's not very understood. And speaking about genetic, you have also a variation, and you don't have studies about the correlation between the genetic and the chemical part in the essential oil. So all of these limit the development of a sustainable production. And the overreaching objective of my PhD is to integrate molecular genetic characterization with phenotypic and phytochemical analysis to better understand the drivers of the genetic and the essential oil variability in Aniba rosodora, and also evaluate the influence of origin on reproductive performance and chemical composition to develop a sustainable rosewood industry. I have 3 main objectives, and I will talk briefly about each one. The first one is more genetic, so we will tracking genetic difference using SSR sequence, which is a microsatellite that I think if you were here before, you heard about it, but it's a small part of the DNA that has a high polymorphism, and it's very useful to detect difference into the species. And we want to use this to assemble a South America collection of Aniba rosodora. So we have material from Peru that I showed you guys here, the results that we already analyzed, and it's very different from the French Guiana material, if you see this PCA. We have material from natural populations in French Guiana, and also from the Risque 2 plantation that I told you about. And we were tracking samples from Brazil. I was there to collect the samples, and now we are going to start analyze. And also we want to use herbarium material because we want to identify if we are talking about Aniba rosodora complex, or is one species, is 2 species. So we will include herbarium material too. These first results, you can see that inside French Guiana, we have a difference in genetic structure. We have one group that's Wanahi, and I don't know if you pay attention, but it's more in the south of French Guiana. And when you do analysis only with these populations, sorry, you have also difference that are noticed here. When you go to structure, there is a genetic software. We found with everyone together, we found 2 clusters. So we have the red cluster and the green cluster that are pointed here as 1 and 2, the results. And when we do the analysis for each cluster, you have the green one staying together. So even if we analyze all of this, the K is 1. So you have São Jorge, Escol, and Combi as one genetic group. And it's weird because one is here, the other is here, and the other is here. But the hypothesis we have for that is that someone took the seeds from here, planted here, and planted here. Because before we don't have like a organization in the plantation before you start planting. So they just took the seeds and plant, and they start a commercial plantation. So that's it's our hypothesis. And when you do a cluster analysis only for the red cluster, you have groups that are geographical close. So that's our hypothesis, is you have a very correlation between genetic structure and geographical position. The only exception is Dunklan, because Dunklan is an isolated community in French Guiana. It's separated by rivers, so we think that the rivers are playing like a genetic barrier in the structure of the genetic structure of the trees. The second question is to explore the influence of provenance, hybridization, and environment on seedlings fitness and essential oil composition. So that plantation I talked to you about that calls Risque 2, I went there and we walk around the block collecting seedlings. The seedlings that they're not used for the commercial plantation, the seedlings that just are in the nature. So we evaluate the regeneration. We have here one plantule that's big and the other very small. They represented different reproductive cycles happening into the plantation. And here you can see a lot of seedlings that we found these groups of seedlings very frequently, and that represents the method of dispersion, because rosewood seeds are dispersed by toucans. And then when you look this and you look up, you have like an enormous tree with a good branch. So we imagine that the toucan were in the branch, they eat the seed and the seed dropped, and then you have a seedling. And that happened a lot because we have a lot of this in the wood. Another traits we were evaluating is height. We have a very good variation, herbivory rate, and also light exposition with the results in accord with literature, because rosewood likes to grow in shadow. So all the seedlings were mostly with 0 classification for light exposition. So we went there, we analyzed all this phenotype data, and we also collect a leaf. And this leaf, we use it half for DNA extraction and half for hydrodistillation, which is we will put the leaves here and we will extract the essential oil. Because the essential oil has a difference in the wield based in plant organs. So wood has one wield. If you use leaves, you have other. If you use small branches, you have other. And we want to understand this configuration. So we are using the leaves in this case, and we will use branches and wood for the adults of Risquetu. And also, we want to know the plant origin we have also the plant origin affecting the essential oil composition. A study made comparing the essential oil from Brazil and Guyana shows that the linalool position is different in the 2 countries. So you have different aromas. If you smell, I forgot to bring it, but if you smell the essential oil from Guyana, it's very sweet and it's like a perfume ready to use. And then if you smell the oil from Brazil, it's very woody. So it's like a masculine perfume and the other is a feminine perfume. But the difference is very, very easy to note. So we want to know that this difference could happen in the provenance level, because we have 6 provenance planted, so this could happen there too. And the third question is develop a predictive model for essential oil yield and composition to inform a breeding program in Guyanese rosewood. So we will do the essential oil characterization combining chemical approaches. We will use gas chromatography and mass spectrophotometry to assess the chemical composition, quantity of linalool and other components, to do models of discrimination, ways to understand how the composition changes in different provenance and different places. And also, we will want to know if the composition could be used for different things, like aromatherapy, perfume industry, antioxidant, or any other medical application. We also use near-infrared spectroscopy, for create a chemical fingerprint that will be used to prediction in situ. So you can use the near in the field, and when you have this chemical fingerprint, you can go there and see, oh, this composition is a good one for this. So I will extract the oil and sell for this proposal. And we also have chemotypes for provenances, so we want to see if we have a chemical difference in the provenance level too. And combining this with genotypes, we want to valorize the essential oil from the different provenances and also create resources for conservation. And the previous analysis that we have in the transcriptomet show that we have different genes in the different provenances. So here is Dunklin, and here is Skull, Lysis, and Wanahi. And these genes are different expressions in the high, in orange, wield, and low in gray wield. So we want to continue this analysis to confirm this correlation between genetic and chemical for the essential oil variation in wield and the composition of linalool. I thank you guys for the attention, and I thank you, my founders, for this opportunity.
Thank you so much. I almost smelled it, you know. Yeah. Okay, so now please welcome Eilish Castillo, who's going to tell us about the integrated climate response and biomass modeling of Picea omorica.
Yes. Well, I hope you can see me because I'm too small for this. Anyway, I'm Eilish Castillo. I'm a PhD candidate at the Nicholas Copernicus University in Turin. Well, so tree rings have told us a lot about climate. But what if we've been only asking half the question? What if those same rings tells us how much carbon a tree gains or loses each year? This time, I'll show you how Picea omorica, a narrow endemic in southeastern Europe, helps us connect these two stories: climate response and carbon dynamics. So, here's the question I kept coming back to while working on this. What happens to species that already lives on the edge when the climate starts pushing harder? In southeastern Europe, especially in interior regions, warming has accelerated, meaning hotter summers, more frequent droughts, and one species particularly at high risk is Picea omorica, or the Serbian spruce. It's not just any other conifer, you know. It's a tertiary relic, as I mentioned, an narrow endemic with limited regeneration and weak competitive ability. So let me ask you, what actually happens when a species like this faces increasing drought stress, particularly in the Balkans? Surprisingly, we don't fully know, right? And that gap is where exactly this study begins. Through collaboration with researchers from Parthenope University of Naples, Italy, we incorporated biomass component, making this the first combined dendroclimatic biomass study of Picea omorica at the Gobsa site in Bosnia and Herzegovina. At its core, our aim was to understand how climate shapes growth and what does that mean for carbon. We focused on temperature and precipitation and how they influence annual ring formation. But we also took it one step further. Can these rings, these thin layers of food, actually tell us something about biomass, carbon storage? Our expectation was pretty straightforward. Hotter or drier summers, meaning reduced growth. And if growth decreases, thus carbon sequestration decreases as well. In other words, can tree rings act not just as climate proxies, but as indicators of carbon dynamics? So, to place our study in the context of existing research, this network shows the major themes in tree ring research. You can see the climate change, drought, growth response, wood anatomy, and even isotopic approaches, all strongly connected. But then, we noticed something interesting. Actually, something missing. Like, where is biomass? Where is carbon or ecosystem service valuation? Why are these only weakly connected or not even connected at all to tree ring studies? And even more, why is there so little data from Gobsa, a site with such a unique climatic and topographic setting? So, this study is not just filling that gap. It's connecting these conversations that have been happening separately. To answer these questions, 3 cores were extracted from the Upper Drina River Basin in Bosnia and Herzegovina with collaborators from the Faculty of Forestry at the University of Sarajevo. So this area is one of the remaining natural habitats of Picea omorica, where it survives in small scattered populations, not forests, but fragments, across roughly 60 to 200 hectares. So we started with 50 samples, but after cross-dating and quality control using Cofetcha, 37 series retained for chronology development and for subsequent climate growth analysis. We then paired these tree rings with climate data focusing on temperature and precipitation, which are the main two drivers of conifer growth. So using this data, we compared year-to-year climate variability with changes in radial growth over a 7 to 1 year period. But instead of looking only at the growing season, we ask, what if the tree remembers more than just the present year, right? So we extended the window, 6 months before the ring forms, and 9 months into the current year, because as we know, trees don't just respond instantly, they carry memory or so-called carry-over legacy effects from the previous year, and as shown also in the previous ecological studies. And once we identified the key climate signals, we reconstructed past climate patterns using calibration, validation, split testing, and residual diagnostics. And this part was guided by training at the University of Arizona's Laboratory of Tree Ring Research, which is the top and largest tree ring research laboratory in the world. But here's where it gets more interesting. We didn't stop at growth. We linked tree rings to biomass using IPCC-based allometric equations. So now the question becomes, not just how did the tree grow, but how much carbon did it store while doing so? Did it stay consistent over time or shift under different climate conditions? And the results, the strongest signal influencing Pizza Omorica's growth didn't come from the current year. It actually came from the previous summer, June to August. You know, when summers are wetter, growth in the following year tended to be stronger, likely because sufficient moisture supports carbohydrate storage. On the other hand, when summers were hotter, growth declined, which just points, of course, to a clear sensitivity to heat and drought stress. So let's think about this. Why would last year's summers control this year's growth, right? Maybe you're wondering. Because of stored carbohydrates, because of late wood formation, because trees prepare in advance to support early wood production in the next growing season. So to make sure these patterns are reliable, we tested the chronology strength using expressed population signal, signal-to-noise ratio, and inter-series correlation. And the takeaway is simple. This is a strong, coherent signal. So when we interpret climate relationships, we can do so with confidence. And in this figure, you see, for example, individual series is compared with the master chronology. So despite some divergence in the mid-century, notably around 1957, so the overall correlation is 0.44, which just reflects the strong agreement and validates the tree's inclusion in the chronology. Then comes with the final climate reconstruction. So the figure you see from 1952 to 2021, what stands out immediately is how strongly Pizza Omorica responds to precipitation, and its growth closely tracks wet and dry periods. So wet periods correspond to stronger growth, so dry periods to declines. And in here we see clear drops in the late 1950s, early 1990s, and early 2000s, which are all linked to major Balkan droughts. So the question becomes, how much stress can this species tolerate before it reaches its limit? Because, under average conditions, it survives, but under repeated moisture deficits, it struggles. So putting everything together, what we see is that Pizza Omorica benefits from stable or wetter conditions. But its growth and vitality drop quite noticeably as droughts become more frequent or severe, especially in the context of future climate change. But this is not the end, because from here, we can connect tree growth with biomass and carbon storage. And since the tree ring chronology is statistically strong, as I mentioned, we can move beyond growth patterns and start interpreting what they mean in terms of ecosystem function, especially carbon sequestration. So in here, when we overlay ring width and biomass, the same drought years stand out, 1972, 1987, 2001, 2012. So years with hot or dry summers, it show narrow rings, then lower biomass. while wider rings indicate higher productivity. So in a very real sense, these rings are not just measuring growth, they are measuring carbon uptake. So using allometric models, we estimated by 2022, the average tree stored about 12.8 kilogram of carbon. It may seem modest for a tree if you think about it, but we can scale it to forest stands across decades. It becomes, highly significant, especially for a narrow endemic like this, which is already limited in its distribution. And from an ecosystem service perspective, knowing this is really crucial because it tells us how much carbon an individual tree contributes to climate regulation, not just how fast it grows. So, what does all of this tell us? Pizza omorica depends on moisture and is highly sensitive to heat. And importantly, changes in tree ring width line up pretty well with changes in biomass and carbon gain, which means tree rings are not just telling us about climate. They also give a good sense of carbon dynamics. So to conclude, this study doesn't just describe how a tree grows, it also shows how growth translates into carbon and how both respond to climate. And as droughts become more frequent, we're not just looking at reduced growth. We may be looking at reduced carbon storage capacity. It's like, a practical framework that can support future climate impact assessments, carbon accounting, and conservation planning, importantly, not just here in southeastern Europe, but as well as in similar forested landscapes. So I'll leave you with this. If trees are natural archives of the past, are we ready to listen to what they're telling us about the future? Thank you so much for listening.
Thank you so much. We still have 2 minutes if there's some questions. No, I didn't see any. Okay, so I may just ask a quick one. If I understood well, you just analyzed one population?
One pizza, pizza.
Yeah? But do you think that if you, if you have more, do you think that you would get different results? Or that would be like,
Absolutely, because right now that's what I'm actually writing in my PhD paper. We are trying to analyze Pinus silvestris, Pinus mugo, Pinus leucodermis, and Pinus omorica, and we're using the water efficiency. So, of course, the result would be different because other species are canopy dominant, and if they're canopy dominant, of course, the performance would be affected as well. So yeah, definitely, and we're looking forward to that, finishing that paper.
Okay, thank you. So if there's nothing more, then please, may I invite Magdalena Terlecka now, who's going to tell us about the effect of sex and clonality on the climatic hydrological response of radial growth in white poplar this time.
So, hello everyone, thank you for being here. So today I would like to show you some results, part of it, about the effect of sex and clonality on the climatic hydrological response of radial growth in white poplar, so in Populus alba. And we found this topic interesting because actually little is known about the consequences of clonal reproduction for the radial growth in dioecious species. And of course, both traits can have a potential impact. So clonality provides significant benefit, especially in difficult environmental conditions. So if we have some environmental disturbances. And firstly, it allows for efficient resource gathering and sharing within the clone. It also reduces the cost of sexual reproduction and, of course, increase the chances of population survival. And in dioecious species, male and female trees can differ in growth rate, in morphology, and research management strategies. And mostly, it is assumed that female trees often have higher reproduction cost and because of that are more sensitive to environmental stresses, especially if we say about water shortage. Okay, so as I mentioned, few studies consider the effects of clonality and dioecious on tree growth at the same time. So there is a kind of knowledge gap in that, and because of that, we want to test two hypotheses. And the first one is that the response to hydrochromatic conditions varies between clonal and non-clonal individuals, and these non-clonal individuals are called singletons. And the second, that female individuals are more sensitive to stress induced by climatic conditions and by river water level fluctuation than male individuals. Okay, so in general, we have two populations, one in Rogaline landscape park and the second in the Dębina forest. So the Dębina forest is inside the city, inside Poznań. And as you can see, there is more than 500 individuals in each population. And we collected 260 cores from both populations. And we aimed to take 2 cores per tree. But the poor health of the trees, especially in the Rogaline Landscape Park, sometimes made it impossible. And because of that, like outside the tree looks really healthy, but when we go into the trunk, sometimes they are just rotten inside. So sometimes it was very difficult to take 2 cores per tree. Okay, but we take the cores, of course, with Presley borer. And after preparation, tree rings were analyzed in the Q recorder software, and further processing and data quality assessment were done in complementary Dendro software. And the climatic and dendrochronological data analyses were performed in R, and this included calculating descriptive statistic and the data detrending. And also we used the tree-claim package to check the correlation between tree ring width and temperature, precipitation, and also between water level in the Varta River and climate data. So the temperature and precipitation were taken from the Google Earth Pro using the crew grid. And information about the Varta River water level came from the Institute of Meteorology archive. Okay, so going to the result, based on tree ring parameters, we found that all Populus alba groups, regardless of sex and regardless of type of their reproduction, create high-quality chronologies, and this can be, lucky for us, used to analyze climate-growth relationship. And the graphs show comparative chronologies of all 3 groups in a studied population. And these chronologies include both rough series and the data that trended using 30-year spline. And in each population, the trended chronologies, of course, had more consistent year-to-year pattern than the rough series. And this reflects the shared environmental signal after removing individual growth trends that could hide it. Okay, so now you can see the graphs with moving 25-year window correlation between tree rings width and, in that case, temperature. On the first look, it seems to be a little bit chaotic, maybe on the second look too, but I try to explain it. So on the graphs, the positive correlation are marked in blue, and the negative correlation are marked in red. And if the relationship is significant, there are some asterisks in the window. And the vertical axis shows months from June of the previous year and to September of the current year. And the horizontal axis show years representing the middle of the 20-year correlation window. And in both studied sites, we found statistically significant links between monthly temperature and the radial tree growth, and the overall effect of temperature was very diverse. So especially the population in the Organic Landscape Park showed more varied seasonal responses because we have positive and negative correlation between growth and the temperature changes. And surprisingly, in the Dembina forest, a consistent negative reaction signal dominated, like in all groups. And additionally, singletons, so these non-clonal trees, reacted more consistently across seasons than clones, which showed more varied direction and timing in significant responses. And the table shows simplified, like very simplified, correlation between temperature and the radial growth. And the results indicate that growth responses to temperature differ between clonal and non-clonal trees, mostly in terms of timing. Okay, so for precipitation, we also found significant correlation to radial tree growth, and it is worth noting that positive correlation were dominated and mostly focused on the current year. And at both sides, spring and summer precipitation in the current year was crucial for the shaping radial growth. And so for Organic Landscape Park, precipitation in June and September was especially important, while for the Dembina forest, July precipitation was a key. And also a shared sex-specific pattern was observed in the studied population. At both sides, female trees reacted a bit stronger to changes in total summer precipitation in the current year than the male trees. Okay, so you can see that clearly that the reactions were concentrated in the current year, and the impact of previous year precipitation was much weaker in case of precipitation. Okay, and this is the most exciting for me because the water level in the Varta River, because both populations were near the Varta River, had the strongest impact on the radial growth among all studied factors. And at both sides, in all analyzed repetition, there was a very strong positive connection during the spring-summer period, so mostly from April to September of the current growing season. And moreover, radial growth in the Organic Landscape Park population was shaped by a wider time frame, which also included the winter months like February and March of the current year. And in both populations, the effect of the previous year was smaller, like in precipitation. And there was also a shared response pattern based on sex for both populations. So male trees at both sides reacted a month earlier. So in Organic Landscape Park, it was January, and in the Dembina forest, it was April. But the reaction of the male individuals in the Dembina forest is not so clear as in the Organic. And you can, there is a clear, the table clearly shows a strong positive influence of the river water level, too much words, sorry, on popular growth. And we find it interesting that the groundwater availability has a stronger impact than a precipitation. And we also checked the draw index. So the analyzed population show a clear and statistically significant relationship between annual radial growth and draw conditions, and that was expressed by the standardized precipitation evapotranspiration index. And in both populations, the correlation coefficients were moderately high and significant for analyzed, and they were significant for all time frames. Like we checked the 6 months, 3 months, and 12 months. And the strongest correlation were usually noted for the 6-month scale. And this suggests that the tree growth in both populations reacts most strongly to the medium-term moisture conditions. Okay, and going to the conclusion. So temperature had a variable effect on radial growth, but in the Dembina population, the effect was strongly negative. We find it interesting, and it might be because of the urban heat island effect, because the population was inside the city. Also, precipitation generally had a positive effect on growth, particularly during the current growing season and with summer months playing a role. The river water level was the strongest environmental factor, showing a clear positive effect on growth during the spring-summer period. And also moisture conditions expressed by SPEI were significantly positively correlated with radial growth, particularly at the 6-month scale. And that's all from me. Thank you very much.
Thank you so much. That was a quick one. We still have like 4 minutes left, I think, if there's some questions. Yeah? Please use mine.
Sorry, congratulations in your presentation. My question is, you said at the beginning about the non-clonal and the clonal, and at the end you didn't give like a conclusion about it. I don't know if it was...
Yeah, I didn't tell the details because I'm still working on it. Yeah, it's fine because we see some differences between the reaction of clonal and non-clonal plants, so the singletons. They are mostly like the singletons share more similar to each other reaction to, for example, changes in precipitation and so on. And the other one are more different. We don't know why. We still don't know why, but we're working on it. And yeah, so I think that's all.
Anyone else? No? Okay, so I may have a quick one. Magdalena, did you see that the sexes tend to cluster together, like they, they form so spatially cluster?
Yeah.
Or they tend to occupy some specific habitats, microhabitats?
Yes. We checked this in our first work, which was about, and the first work was about population genetics. So mostly the female individuals are grouped like in the valleys when the water is more available and there was more of nutrients. So that was similar to our hypothesis that they are more fragile. So they need the place where the conditions are just better. To answer your question.
Yeah, and how do you think this influences your results or the interpretation?
Yeah, if the female individuals are in better conditions, so they are better times to grow. But anyway, they are more fragile to changes in temperatures and precipitation and so on. So this is. I don't know.
It's not enough for them still.
Yeah, there is not. They just female individuals just need more, all things.
Okay, thank you so much. Okay, so now let's move to the next presenter, Sergio Pedro Jr., who's going to tell us about thermal plasticity and maturation-induced dormancy in Anadenanthera macroparpa seeds, the species which personally I don't know, so I'm happy to find out.
Okay, so good afternoon, everyone. My name is Sergio Pedro Jr. I'm a PhD student in Brazil on the State University of Londrina, and I'm doing a fellowship here in Poland in the Dendrology Institute for 6 months over Professor Mikołaj supervisor. And today I'm going to present for you the thermoplasticity and maturation-induced dormancy in Anadenanthera macrocarpa seeds. Well, for beginning, I think it's important so you have some knowledge about Brazil itself. We have 17% of the world's flora, so we are the most biodiversity country in the world, and one of the biggest ones. We are also, Yeah, we are the only country that are cut by the equatorial line and for the tropic at the same time. So we have a lot of differentiation in the temperatures and the climate itself. So this is one of the reasons that we have so much different species in there. And a lot of these species, since we have so much, they are understood. They are highly understood species. For example, last year we have more than 900 new species described. So it's almost 3 species per day. So we have a lot of knowledge that we still need to do on these seeds, on these species, for you can propagate them and put them in the commercialization, as well as to preserve them. And we also have a lack of standardization germination protocols for these species. This one that I'm presenting to you today is already used in restoration protocols as well as used in the landscape. So we are already using them, but we don't know how to use these seeds as well. So this is part of the aim of this project. And it's important for the sustainable ecological restoration, so we can have seedlings more vigorous than other ones. As I said before, it's a native species from Brazil and have a lot of ecological it's an ecological important plant. And it's a keystone for restoration because it's from the dry lands, so it can live very nice in dry stress spots. And it's one of the challenges is limited by understanding how maturation and temperature interact to regulate the dormancy of these species, because we still doesn't understand completely this one. Just for other curiosity, here in the north part of Brazil, we are closer to Canada than to the south part of Brazil. And here we are close to Africa than to the other part in Acre. This is the species Ananderantera macrocarpa. This is the tree, this is the flowers. And here we have fruits that are still getting to the full maturation stages. And as I said before, we have different uses for this species. We have the ecological uses, is a fabaceae one, so we can fixate nitrogen in the soils that are devastated. This is one of the modes that they are used in the restoration protocols. The other one is because it grows faster, so it's a good species from this. It's also very resilient because it's native from the drylands. But we also have some economical and biochemical value in this species. As, for example, it's rich in tannins that have a lot of antimicrobial and anti-inflammatory properties in it, as well as used to do nanoencapsulating process in some compounds that normally we use for oxynitric compounds. As well as the timber is also used in the South America because have a high density and it's pest resistant. And here we have a map from Brazil and the areas of distribution of this species. As you can see, it's well distributed in all the country because we have different biomes that can have dry lands in it. It's normally in the Caatinga and the Cerrado biome, but we also have rupestриan and altitude camps that can have these species. For example, this research work carried here in the Paraná state, the one that I live and do my master's degree. And our aim was evaluate the germination behavior, vigor, and dormancy patterns of mature and immature seeds from A. macrocarpa seeds, subjective to suboptimal temperature, moderate temperature, and supraoptimal temperature. That was 10 degrees, 30 degrees, and 40 degrees. Well, the experimental design. At first, we selected 2 different stages of maturation from the fruits by physical qualities. We use fruits that was green and fruits that was brown. This is the way that we separate the immatures from the matures one. And we use these three temperatures that I already told you. So here we have an image from the fruits, the completely mature ones and the immature ones. And here we have the seeds in the two different stages as well. As you can see, they have different sternomorphological characteristics. Here we have the paper rolls. We did the germination test. We use these paper rolls because they give us like big seedlings, so we cannot germinate them on Petri dishes or in jar box. And then we weigh the paper that we are using and put 2.5 times the weight of the dry paper from water, so we can have enough water for all the experiment and don't need to water them again. Here it's one more image from the fruit and the seeds, the immature ones and the mature ones. One more image from the seeds after the beneficiation process. And now the results and discussions showing the contrasts in dormant patterns into the maturation stages. Well, for starters, the mature seeds, they doesn't germinate at all. They have adapted dormancy, so they have a deep physiological dormancy. And this is adapted strategy to avoid extra dry because they are from the dry forest, so it's an ecological approach from this species, and ensure survival by prioritizing long-term establishment over immediately growth. So these species normally have dormancy in the seed, so it can be armazenated into the soil instead of germinating after it falls on the floor. This is the data from the immature seeds. From the green seeds, we have the best temperature for germination being the 30 degrees with 69% of germination, followed by the 10 degrees with 60% and the 4 degrees with 10%. The 4 degrees, when we are now studying the denaturation of the proteins from the seeds. This could be the problem we have from they don't germinate it. And here we have a synchronized pattern from enzymes activity because they have a bigger germination speed index, as you can see here, followed again by the 10 temperature and the 440 temperature. This could look like a simple study because, as I said before, we have a lot of different species, and we need to first establish these protocols for germination over doing some more complex researches, because otherwise we don't have seedlings enough to do this more complex researches. And as we see in this study, Amacrocarpa seeds germination is strictly modulated by dormancy status and temperature. And when you have completely formated seeds, they don't germinate yet. They have physical dormancy. The other conclusion is the moderate thermal regime, that's 30 degrees, favor optimal metabolic coordination. And in the 31, deep physiological dormancy in mature seeds is a key evolutionary adaptation for dry forest. We are still looking to see if this dormancy is physical or physiological, because the seeds was heavier after the germination test, even the ones that were not germinating. So this could show they have some imbibition doing it there. But when we try to do the tetrazolium test without imbibing the seeds again, because they are already imbibed in the paper, they don't show high viability. This could indicate they have some imbibition, but the water was not enough to germinate the seeds. So thank you, everyone. This is my email in case everyone wants to have some doubts.
So, any questions? No? Yeah. Okay, so I think it's gonna be me again. Is there a good way to precisely tell at which developmental stage your seeds are? Because when you differentiate them, just like you have immature and mature, and you do it mostly visually, then I'm afraid that among these immature, you will have, you know, some that are more mature and some that are not. Is there any good way to do it in a better way or more precise?
Well, we put these seeds into a scanner, a ground eye scanner, so we can have an RGB qualification from the color of the external morphology from the seeds. So we have like the data into the how green they supposed to be immature and how brown they supposed to be mature. So we can separate them by the color, not only from the fruit color, but also from the seed color itself. Yeah, then we just rip the fruit, beneficiate them, take out the impurities, and then we separate them according to the color.
Is there anything more? No? Okay, so I'd like to invite the last speaker, I should say, but not the least, of course. Please welcome Ocean Williams with trait variation across multiple biological scales in recalcitrant fagaces seeds.
You want to point to it's here.
Okay, I'm Ocean Williams. I'm a science intern at Kew Gardens over in the UK. I'm currently studying for my bachelor's in biology. And today I'll be talking about trait variation across multiple biological scales in recalcitrant seeds, specifically oak and chestnut seeds. And this is done with the help of Namrata Pradhan and Louise Colville, also researchers at Kew Gardens. So to give an overview, it's been covered by quite a few presentations, but recalcitrant seeds are sensitive to drying, essentially. And to describe these seeds, seed traits are used, well, seed traits are used to describe any seed, things like mass and moisture content and so on. But these traits are often fixed at a species level. So this study is going to examine how these seed traits vary across different biological levels. Specifically, the species I'm focusing on, we have 4 different Quercus species, which are oaks, and that's Quercus rubra, Quercus roba, Quercus ilex, and Quercus petrea. And then we have 1 Castanea species, Castanea sativa, and that is sweet chestnuts. Okay, so for the methods, we collected the seeds from the Kew Gardens Wakehurst site in the UK last year in autumn. We tried to collect them as close to their natural dispersal point as we could. So we would lay a mat down underneath the tree and then shake the branch with a hook and take the seeds back to the lab. There was some kind of preliminary screening at this point. If the seeds were visibly damaged or germinating or cracked or anything, we wouldn't collect those ones. So in terms of the seeds we collected from multiple trees for 2 of the species. So for Castanea sativa and Quercus roba, we collected from 6 different trees, just for those 2. The other Quercus species, just because of what was available to us, we could only collect from 1 tree in that case. Following this, we selected 10 seeds at random per tree, and that includes the 6 different trees for the Quercus roba and Castanea sativa as well. And then measured a range of seed traits. So we started with mass. We first measured the fresh mass of the whole seed, and then we dissected the seed into different tissues, the cotyledons, pericarp, and embryo, and we measured the fresh mass of those. And then we put them in the oven at 100 degrees for 17 hours and measured the dry mass of all these separate tissues. From this, we were able to calculate the moisture content as a percentage of both the whole seed and also the individual tissues. And we could also use these dry masses to calculate the seed coat ratio as a percentage rather, so how much of the seed is made up of the seed coat. Then we looked at how these traits vary across these different biological levels that I've mentioned. So starting, this is in the figure on the right there, starting at the sort of top hierarchical level, we looked at interspecific differences in these seed traits, and this was only across the Quercus species, so not the Castagna at this point. And we just looked at how these seed traits vary across these different species. Following this, we looked at, this is where the 6 trees from Castagna sativa and Quercus roba come in. We looked at the intraspecific differences and how these seed traits vary across different trees of the same species. And then we also wanted to look at whether or not there's a lot of variation within one individual tree. So of the 10 seeds we collected from a tree, how much did they vary from each other? And the final level was at a tissue level, specifically looking at the moisture content differences across the pericarp, cotyledons, and embryo. And this is just a cross-section of a Castagna sativa seed, just to show you the different tissues that I'm talking about. I don't have this for the Quercus species, but they look relatively similar on the inside. Okay, so for the results, starting at that top level, the difference in the Quercus species. This doesn't show all the results, this is just some of the traits we measured. And I'll talk through the setup of the graphs first, because it's going to be the same for the next few slides. So on the top row, they're all mass-based traits, the top left being the whole seed fresh mass, and then the middle top is the whole seed dry mass, and then in the corner is the embryo dry mass. And then underneath, we have a couple of the moisture content traits, with the bottom left being the whole seed moisture content, the bottom middle being the embryo moisture content, and then finally the seed coat ratio in the bottom right corner. And this is showing all those different Quercus species that I mentioned before. So from these results, excuse me, we can see that most of the traits differed between the species, but not all the species differed from each other. So what I mean by this is, for example, if we look at the mass traits on top, you can see Quercus ilex was significantly smaller than the other species, whereas some of the species like Quercus roba and Quercus rubra, they were not significantly different from each other. And this was common with a lot of the traits that we looked at. So we found most of the variations in the mass-based traits, and this can be seen in the graphs. With the moisture traits on the bottom, we found that there was actually no significant difference between any of the species for whole seed moisture content. However, embryo moisture content is where we actually saw most of our differences, with Quercus ilex having significantly more, a significantly higher embryo moisture content than all the other species. And then finally, seed coat ratio. You can see the clear outlier is Quercus rubra, has a very, very high seed coat ratio compared to the other Quercus species. So this is just showing that first level of variation and how the different species vary from one another. So to go a step down, and this is just looking at this point at Quercus roba, at those 6 trees, the 6 different Quercus roba trees. And the setup is the same for the graph, so the mass traits on top with the embryo, the moisture content traits, and the seed coat ratio on the bottom. So in this case, we found that the mass traits showed the clearest differences. There was a massive range of variation depending on which tree we collected from. And it was, the most variation was in the whole seed mass traits. There was slightly less significant differences between the trees of the embryo mass, so implying maybe there's more consistency in embryo size across the species, but there was still quite a lot of variation, which you can see on the graph. And then moisture content traits showed slightly less variation. Whole seed moisture content did have one clear outlier, and that was tree 4. There was a little bit of variation between the other trees, but with embryo moisture content, we actually found no significant difference across any of the trees. There was a lot of overlap there. And the same goes for seed coat ratio. There was quite a bit of overlap, apart from one outlier. I think it was tree 1 had a strangely higher seed coat ratio. So that is looking at the differences between different trees and the seeds they produce. However, with this graph, you can also look at the variation within a tree. So because it's a box plot, you can see the range and the amount of variation that there is in any individual tree. And you'll see that in the mass traits, not only between trees, but also within trees, there is quite a lot of variation. And this tends to be a common theme, that there's just a lot of variation in the mass-based traits, especially compared to the moisture content traits, where there seems to be slightly less. However, it's still significant enough that it can make an impact, because with these recalcitrant seeds, things like moisture content is so critical. So even if there's a little bit of variation, it does make a difference. Okay, next is the same setup, but for Castanea sativa. So similarly, we found that the whole seed mass-based traits showed a lot more variation than anything else. And it was again the same trend of slightly less with embryo, just the embryo mass, but still there were some significant differences. Where this does differ from Quercus robur is that if we look at the embryo moisture content, there actually was quite a lot of differences between the trees, significant ones that is, which was different to the Quercus robur results. And also seed coat ratio showed a great deal more variation within a tree as well as between trees when compared to the general trend in Quercus robur. And then the final level, we have the tissue level variation. So that was looking at the moisture content in the embryo, cotyledons, and seed coat. So I've displayed this only as one graph. This is including all of the species, all of the seeds, all in one, because after analyzing everything separately, we came across the same consistent result that the embryo had significantly higher water content than the cotyledons, which was significantly higher than the seed coats. And so this shows that there is this distinct separation between all the tissues at all biological levels across all the species in the moisture content of these tissues. Okay, so interpreting the patterns of these variation, we can say that variation occurs across all these different biological levels we looked at. So variation is not only between a species, but is also within a species and even within a tree. Mass-related traits, they tend to show the greater variation, and we can also conclude that moisture content differs consistently between these different seed tissues. And we found that these kind of patterns are broadly consistent across species. So why is this important? So these seed traits can often be summarized per species, but because we can see from this, trait values vary widely within species and within individual trees. So these species level averages may not capture this full range of variation. You can see in the image, these are 5 of the 6 Quercus robur trees we collected from. Each row is a tree, and you can just see visually how different they are from one another, or even the differences in size within some of the trees. So this is particularly relevant for recalcitrant trees where, as I said before, traits like moisture content are critical, and variation in these traits may influence how these seeds respond to storage and handling. We could also say that the tree level variation may influence which individuals contribute to regeneration. Applications and future work, though I should really say current work, all of this is ongoing at the moment at Kew Gardens. We're doing embryo cryopreservation trials and germination tests. So using the embryos of these seeds, we are freezing them in liquid nitrogen to try and figure out a way to store them. We're tracking individual embryos and traits across trees. So we're doing this tree by tree to see if there's different responses based on the tree. And we aim to identify traits which might influence cryo success and therefore hopefully support the development of cryopreservation protocols for these recalcitrant species. So to conclude, we can say the seed traits vary across multiple biological levels, and much of this variation occurs within a species and within trees, not just between different species. We can see different traits show different patterns of variation, and perhaps seed traits should be interpreted as ranges rather than fixed values. That's everything. Thank you.
Thank you so much. And I think at this point I can already open the discussion. So if you have some questions to our last speaker, then yeah, go ahead.
Hey, good afternoon. Nice presentation. I'm interested in the cryopreservation part. You said it before. Do you know how low the moisture content should be in the embryo so you can cryopreserve them? And do you use some VPS solution or some encapsulation process in the embryo so you can cryopreserve them?
Yeah, so currently what we're testing is the best moisture content. So far, we've not really seen much success. This might be because we haven't actually used any cryoprotectants. Namrata, who is the colleague I mentioned earlier, who's organizing these studies, she wants to look at using PBS2 or some kind of vitrification techniques because we're just not really seeing much success. But we're testing a few different water contents. We range from 0.4 down to 0.2, and we're testing this for all the trees. But it's ongoing, so we don't really have much results yet.
Can I have a question? I was wondering, you showed that Quercus in comparison with the whole moisture content of the seeds, the embryo was very moist, around 80%. Right. Yeah. And do you maybe know why? What results in such a big difference? If there's like difference, the seed coat, how it's built, or anatomical reasons on.
Yeah,
what do you think?
So I hadn't actually considered this. I've not really given much thought into why there is this variation. I've just sort of been capturing that it exists. But yeah, in terms of the Quercus ilex being so high, I really don't know. I can't think of why. Yeah, I'm afraid I don't have anything for you. I don't have anything, but it was a good question.
There's something to consider in the future, maybe.
Mm-hmm.
Anyone else? Yeah. Go ahead.
Thank you for the presentation. It was very interesting. I was wondering, in the context of invasiveness of red oak in Europe, have you thought about it, and maybe you considered how these factors that you have studied can influence its reproductive success in Europe?
I didn't know it was invasive in Europe, so I have not considered about the wider implications of this. Yeah, again, I've not really given much consideration beyond the capturing the variation itself, and specifically I've been focusing on the links to cryopreservation. But yeah, I don't know, it's definitely something that could look into in the future, but I don't have any consideration for that at the moment. Thank you.
Anyone else? No? Okay, thank you so much. And I think we can go back, starting with the first speaker of the session, about rosewood. I have a question. Will your results be used somehow, like formally, to protect the plantations, or to have it like more sustainable production?
Thank you for your question. We are going to use as a way to help the producers. If we identify different provenances and the essential oil variating and be unique for each provenance, we will help them to sell it as a green certification. And we're also going to see if the genetic diversity is being impacted by the commercial plantations. We are monitoring the natural population surrounding commercial plantations, and we want to donate all the data we obtain to the administrative part of French Guiana that takes care of the conservation, to help them to choose areas to conserve and avoid the plantation in their area and things like that. So yes, we are going to spread our results.
Nice. Okay.
May I speak in Portuguese and after you translate? Fernanda, você acha que a diferença entre o óleo da semente e o cheiro da semente do pau-rosa do Brasil é diferente da Guiana, porque do Brasil é população natural e da Guiana é plantio, pelo que você explicou, né? Isso, você acha que a diferença é isso? Porque o linalool não muda, mas o cheiro você falou que muda.
I will translate the question first. She asked me if there is the difference in the linalool and the oil and the oil aroma is because French Guiana is commercial plantations and Brazil is natural population. And no, because we also have commercial plantations in our sample data from Brazil, and we also have natural populations in our data from French Guiana. So we wanted to know how much the genetic play role in the essential oil variation, and how much the environment also play. Because linalool, it doesn't change the composition, but it changes the percentage of linalool and also change the position of the isomery. And the position of the isomery is responsible for the different aromas.
Okay, thank you.
And my question is regarding, because you said that those plantations are usually established with seeds, and if you will be able to determine the best individuals, best populations with very good concentration of this oil, is it possible for this species to propagate vegetatively, so you could have exact varieties with enriched oil for commercial use?
Thank you for your question. Yes, in Brazil they are already trying to do vegetative propagation of rosewood because it's simple. And the seeds are not always good to plant and produce seedlings. So yes, they are trying, but I don't know and how much is better to use clonal than material from seed, because you have climate changes and you have a lot of things happening too. So yes, I think you can try, but if we confirm that like the unique aroma and unique oil from this DNA, we can try to use like control pollination to certify that this oil this plant will be from this material that had this oil. So maybe, but you need to be careful.
Thank you very much.
Anyone else? No? Okay, so let's move to the second speaker. Irish Castilla and Paisia Morica. Don't be shy, just shine. No? Okay. Okay. Yeah.
If there's any particular reason why you choose Paisia Morica for your species of study?
Well, I'll be honest, because Paisia Morica, we were working with our archival increment course, so that was the most convenient species that we had at that moment, because we were rushing and I was in Italy at the time on my Erasmus and really needed species to sample with. So it's an increment archival course.
Okay, I don't see anyone else, and I've already asked mine, so let's move to Magdalena Terlecka and White Poplar. Yeah, go ahead.
I have a question, Magda. There was a question about this clonal and non-clonal, but how did you recognize them?
Okay, that's a good question. I didn't mention it. So the first step in our project was to was the population genetic analysis, and part of that was to collect the leaves, and after that isolate DNA, make PCR and sequencing. So I did all that thing, and that allows me to check which genotypes are shared by individuals, and that was the clones, and which genotype is individual, so there is nothing more like that. So that was the singleton, so the non-clonal band. Yeah, so that's how I identify which are clones and which are not.
Magdalena, do you remember the clonality level in the two places?
Yeah, I did that, but
not precisely. Just if
yeah,
it was huge, and
it was a lot. Like I think that we have, for example, in Rogaliński Landscape Park, we have 174 genotypes, and the individual, like the singletons, were I think maybe 40 or something like that, and the rest were clones. So the most of the population in both sides, so in Rogalin, in Dębiec, were clones.
And was there a difference between sexes in clonality?
Not really. We are surprised, but not really. That they are like half to half. Yeah, that's right. So in Dębiec, if I remember correctly, they are just half to half.
And the sex ratio, just like in general.
There were no significant differences.
Okay, yeah. Perfect population.
Somehow,
for now. Yeah, okay. Yeah. Could you pass the microphone? Yeah.
So you showed a video where the Jews were coming out, and what to do in a situation like that? Can you drill, or you have to wait, or you can't take the sample anymore?
So we try to take the samples anyway because we are in the field and we have not unlimited amount of time. So we just take the cores. Sometimes there are like in 3-4 pieces, and we try to glue them the best as we can. But they are wet, so we have to wait to dry them, and they are really smelly because the tree inside was rotten. So we wait until it was dry, and after that we glued it and grind it and make all the procedure. But it was harder. Like in Dębiński forest, in the second population, it was much easier because we didn't have like sick inside trees and that. And the Rogalin was special, just special.
Okay, thank you so much. I don't see anybody else. So now, any questions to Sergio about Anadenantera macrocarpa? Sorry. Yeah. No. Oh, okay. There's one question.
Sorry, congratulations in your presentation. My question is: do you think that you don't have any germination in the maturation, and after you explain they need more water? Do you think this is related to the origin of the species from like a dry biome and things like that? And maybe they just need more time to germinate?
Well, I mean, maybe it could be correlated to the provenance of the seed, because I use some seeds from the Rupestrian forest in Brazil, and it's like these species have some phenotype. This special population have a different phenotype from other ones. And if you look to the literature for like previous studies doing with these species, they don't have dormancy in some providences, they have dormancy in other providences. So maybe it could be something related to the genetic population itself, not necessarily to like less water than they need to, or something like that. Maybe it's about the genetic part.
Sergio, are you going to do some other kind of analysis with the species specifically?
Well, this specific research that I showed today is it's from one of my scientific initiation bachelor students, so it's not part of my PhD itself, because in the moment I'm studying crop seeds like wheat and corn bean and soybean. So we are doing another analysis in this species, like cross analysis, and we are trying to do some X-ray analysis in the seeds as well to view the viability from another angles. But in the moment, we are like this specific species are like, we are not working with that in our lab anymore, but we still have seeds storage. So we can do other analysis, but in the moment we are not doing it.
Okay, thanks. Yeah.
But would it be interesting if you study further what kind of dormancy it is? Because you wrote deep physiological dormancy, but I'm not sure on what basis you claim that is this exact type of dormancy, because it might be also different, like you mentioned at the end.
Yeah. We wait the seeds before we put them to germinate, and then we wait them again after finalizing the germination test, so they are heavier. So so we think they absorb some water. So we don't think they have physical dormancy, like at least not like a cold problem that they are not being able to imbibe it. So right now we can do some further analysis. Maybe we are thinking about doing some kind of anatomy in the seeds, but since they are very tough, it's not so easy to do anatomy in very thick seeds. So again, the seeds are there storage, but we are not studying them in this moment.
Thank you very much. And one side note, I'm not a professor. I'm very far from it.
Okay, I guess this was the last question. Yeah, okay, so that closes our session. Please give a round of applause to everybody. Thank you so much. Yeah, and now we have like 18 minutes till dinner. Yeah, so thank you once more, and have a nice evening, and see you tomorrow.
Yeah, one thing about tomorrow, we meet all each other 8:15. But please be packed already. You will store your luggage here in the hotel. But when you're gonna come back from the institute, from the workshops, you need to be ready to depart to the Wolin National Park. So you need to pack in the morning. Yeah, you're gonna check out. You're gonna leave the luggage here, go to the institute, go back to the hotel for lunch, not dinner, lunch, that's true. And then you get after the lunch, you're gonna take your luggage and go to the Wolin National Park. Yeah? Simple enough.